A very good morning for all of you. Ladies and gentlemen, welcome to the day 2 of the 12th International Session, an international conference of Sri Lanka Association for Laboratory Animal Science, themed toward One Health, responsible and reproducible animal-based research in support of precision medicine. Before starting today's session, we would like to remind you all, especially our student participants, that we are going to continue the quiz contest where you can answer a question based on the session that we are having. So you still have the opportunity to win a prize from Sri Lanka Association for Laboratory Animal Science. So hope you will listen to all the sessions more attentively. With that notice, to begin today's session, I would like to call upon Dr. Renuka Tilaka to introduce our first speaker of the day, Dr. Patrick Sharp, Executive Director of Veterinary Services at the University of California, USA.
Good morning, everyone. It's my honor to introduce Dr. Patrick Sharp and to read his citation. He has been a great friend of us, SLALAS. Dr. Sharp is the Assistant Vice President for Animal Resources, attending veterinarian at Purdue University of Laboratory Animal Program and Clinical Professor in the Department of Comparative Pathobiology. He is a diplomat of American College of Laboratory Animal Medicine. He has global laboratory animal experiences in Asia, Europe, Oceania, and North America. He contributes to anesthesia and analgesia research towards enhancing clinical pain management research and providing educational opportunities about anesthesia and analgesia. Dr. Sharp has planned and developed Vivaria and research support space in many areas of the world. He has helped numerous institutions pursue and achieve a ALAC accreditation. With that very brief introduction, I would like to invite Dr. Patrick Sharp to address the gathering. Over to you.
Thank you. All right. Beautiful. All right. So thank you, everyone, for attending today. It's a really important topic. So last night I went in and I redid my presentation based on what I heard on the first day. And I think the reproducibility crisis, it's billions and billions of dollars that are wasted. And I think there's a few things that I just wanted to talk about. So originally, myself and John Schofield, who is from New Zealand, he and I worked on a workshop in Melbourne in 2018. And we started putting together bits of this presentation, but then it's amazing how many different, and I'm only going to touch the surface. I probably have 150 slides, but I'm only going to go through a handful of them. There are some folks that might need some zebrafish help or want to consider the Zebrafish Husbandry Association. So please scan the QR code. And it's a great resource. They have a podcast. They have a Friday Zoom session, which would be a Saturday Zoom session for folks here in Sri Lanka. And I think it's a great opportunity to kind of learn more about zebrafish and kind of see that a lot of people are having the same problems globally. So with research variables, they come into several different categories. So I think some of those are due to compliance. And related to compliance, we have animal rights, but I've separated those out, and I'm just going to touch upon some of those briefly. There are intrinsic items, which are related to the animals themselves, and then extrinsic matters, which I'm just going to touch on a handful of items, which would be things like antibiotics, anesthesia, and those type of things. And then there's another group that I put in as crazy, which is related to things like earthquakes or other things which impact us worldwide. So research variables are frequently overlooked ways to kind of manage those items. Our professional judgment and the science. And that's the benefit of having an organization like ALAC. ALAC is a strong advocate of not only your professional judgment on these things, but also if you are not doing something according to, say, like the guide for care and use, if, like, for instance, cage changing, if you go to an extended cage change, they want to make sure that there's some science behind that and that your ethics committee or your IACUC has reviewed those changes that you might be advocating. So for compliance, we've talked a little bit about, I think with that you have the rules and regulations, which are kind of centrally managed. They might be national or subnational. Institutional policies and guidelines. So an institution might develop some SOPs or some guidance documents on things. And then there's voluntary compliance. So one of those, and these are voluntary, so ALAC accreditation and the ISO 9001. I've worked at organizations that have pursued ISO 9001 2015. So I think these are all great things. And I'm not anti-ALAC. I'm just simply saying that this is a voluntary accreditation. I think it's great to have something to benchmark yourself against other institutions, both nationally and internationally. Briefly on the research variables, these come in all kinds of different flavors, so they could be things like the microenvironment, what's going on in the cage, the macroenvironment, the room or the structure itself, the mega environment. Frequently you'll have many different things going on in the buildings. Not always we have a dedicated vivaria that's dedicated to one building. Materials or items that are going on within the campus. And then we live in a global environment, so we're getting animals from overseas, or we're interacting with colleagues internationally. So how are we doing the type of research that they're doing to make sure that we're optimizing everything? So some of these fall into the rules and regulations. We talk about the cage and the primary enclosure, and I've got some slides to show about that later, but there are guidelines on that. We talk about the temperature and humidity, not only of just the animal holding room, but where we're storing our food and bedding, that those should be within a certain humidity, within a certain temperature to prevent the degradation of them, gases and particulates, and there could be some metabolic changes if we have high levels of ammonia or other types of gases that are in the cages. So with cage sizes and densities, those may be set by laws and regulations. But an organization like ALAC, they would permit you to go beyond that, depending upon what data you provide. Like for our institute, at one institution, we permit trio breeding. So the cage densities, there's a school of thought thinking that by having fewer mice per cage, that we may be advocating that there would be more aggression in some of these animals, and there is some data to back that up. So this is a group that looked at different cage sizes and densities. They looked at black 6 mice, and they kind of went above and beyond with what was on in the guide, and they found about the urinary testosterone level in the BALB-C mice, which can be an aggressive line. And they basically looking at those aspects that affect the housing. This is a group out of the Jackson Laboratory. Everyone's probably heard of the Jackson Laboratory. And here they found that mice housed in individually cages for up to 9 months at twice the density had no adverse or any measurable effects. So all of this data predated the 2011 version of the guide, but it was never incorporated into the guide. But many institutions are going on and providing a higher density. Looking at the macro environment, so the room structure. For those of you who were last year, I talked a little bit about different vivaria. So, this room holds the cages. What's the ventilation? We heard about some challenges that some institutions have had with ventilation and ventilation capacity. Obviously, temperature, humidity, lighting are an issue. The great thing about, I lived in Singapore for several years. We could have windows in the rooms because our light cycle was on in essence, a 12-12 light cycle being on the equator. So you can't always take advantage of that. With the building itself, you have to look at how that building is designed, it's laid out, the different components, where the vivaria is located. So if the vivaria is located in the basement and there's lots of construction going on, or you're near a major highway, that vibration could interfere with breeding or some of these other animals. So I think those types of things are very important. And then, if a shared building creates some challenges. So I think you should be aware of those as you're planning buildings or looking at buildings or looking at different activities. The Giga environment is kind of the campus or the institution. And with that, we look at the animal care and use program. So the ACUP is the animal care and use program. And there's this model of a three-legged stool. So there should be an institutional official, there should be an animal ethics committee or an IACUC, and then there's attending veterinarian. And these items should be in balance. And if they get out of balance, then institutions tend to have problems. And those problems could be related to finances, providing political support, or just general support to the program or the veterinarian themselves. And sometimes some of these programs get really tied up in very tiny things or minor things. So I think it's always important to make sure that you're looking at the big picture. And that's where a place like an organization like ALAC comes in handy, because every 3 years, to me, it gives a way to refocus the program, to look at things, to see how the program itself could be better. And then the terra environment I think the big thing that we see is animal trading, but we're also seeing that with zebrafish now, with them being swapped around. And with that, there comes the potential for infectious disease, a lot of other transportation and regulation issues that could be impacted. So regulatory variables, one of the problems we see is that regulations frequently fail to address things like professional judgment. Those are frequently put in by voluntary accreditation organizations. And the other problem we have is the science. The science changes so rapidly. I think there's roughly every 18 to 24 months, the volume of scientific information doubles. So the problem there we have is the science doesn't keep up with the regulatory environment. And I think that's one of the struggles that we're going to see as they develop the new version of the guide. There's also self-imposed regulations. So animal ethics committees come up with policies and guidelines, or we come up with SOPs. So making sure that regulation, that internal regulation, doesn't become too onerous. And this is a paper that was published in FOSSEB Journal in 2014, and basically just talking about the internal compliance bureaucracies that are within institutions and how sometimes those can become overly onerous. Talk a little bit about animal rights organizations, and these add a whole different layer to your, or can add a whole additional layer to your organization. Things like security, freedom of information requests. These are all things that became very popular during COVID in the US and some other countries. I think one of the items that, a lot of these protests, we see a lot of black and white photographs. I mean, we all have iPhones. My iPhone is in living color. So there's a lot of these photos, and a lot of this information is quite old. I think the other thing that's really important is we're seeing, and I think, Suresh, you talked about this, we're seeing a lot more openness and transparency, and I think that's removing the argument, and I think that's the better way to move forward. I think initially there was a concern, is this a Trojan horse? Like, are we opening ourselves up? But I think we have to open ourselves up. And I've even seen this with even inspection reports from regulatory bodies, that they've just published those, and at least it gives them a way to kind of say, yeah, we got this wrong, this is what we did to fix it, and this is what we're going to do to prevent it. And I think that's a positive thing. One of the things that really has hit is, especially with primates, and will also impact mice and rats as well, is the shipment of these animals around the world. It's a global marketplace, and we sit there and say, well, okay, well, if they don't fly, are you going to truck them everywhere? So to go coast to coast in a big country like the U.S. or Australia, I mean, we're talking 5 days to get coast to coast. And what if you're coming from Asia? Because that's where all the primates are from. So are you going to put them on a boat? I mean, that's 6 weeks. So that's kind of ridiculous to kind of think about that. And what is the animal welfare issues that come into play for an animal that is going to be in a boat for that long? I kind of dug into this a little bit. So back in 1935, probably no one here remembers that, it took 12 days to go from London to Brisbane. 12 days. That's by plane. So you would only fly during the day. There was all these other bits that went on. I mean, it's hard to believe. Recently, there has been a Perth to London flight offered by Qantas, and that replaced the flight from 1947, which was a 4-day journey. So, I mean, by putting, after World War II, there was a lot of air transport of primates. That's really opened up biomedical research and really gave us the cure for polio. That wouldn't have been possible without things like ketamine and air travel. So intrinsic variables in the animal, and I just kept this very simple, and really this comes down to genetic monitoring. For people that are doing a lot of breeding colonies, make sure that you have the exact background strain. A lot of times you'll see in papers, oh, it's a black 6. Well, what does that mean? Is it a black 6J? Is it a black 6N? Is it a black 6 taconic? You're going to get huge differences in research results depending upon the type of black 6 that you have. And then make sure that you understand the breeding protocol. I think this really became important when we saw the human ACE mice that were used during COVID and to study COVID. And I think that those breeding protocols were a little bit more complicated. They got easier as COVID went on, but I think those are important, that you understand the breeding protocol and you know what you're doing. For genetically modified mice, I always tell people, when you receive those animals, check the background strain. So there are tests to do black 6 or whatever. And then you also want to make sure that to do the gene of interest. And I've been involved with places where we've had reputable vendors and the wrong mice got sent. And you don't know that. But if you're 6 weeks or 6 months down the line, then you really could be in a world of hurt, because you have a lot of research that's really dependent upon those animals. But even simple things like looking at the litter size. So if I told you, hey, these were litter mates in a black 6 colony, I think everyone would say, wait a minute, what is this mouse doing here? So look at the litter size, because the litter size tends to be larger. If the litter size changes, that could indicate that you have some breeding problems. And if the coat color changes, that could also indicate that there's a problem as well. I highly recommend that people look at this. So if you're maintaining your own breeding colonies, Jackson Laboratory has several different webinars that are available on demand. So and about a lot of different topics. So I think education is one of those things that we can do to kind of minimize a lot of research variables and research interference that might be occurring. Extrinsic variables, there's a lot of different general articles on these. So Corey Brayton has this in Veterinary Pathology, and she's kind of alphabetized all these problems and the different references that are there, which is very helpful. We give antibiotics to animals. And with that, we see that there's delays in collagen. Here's a study where they're looking not necessarily at what the antibiotics are doing, but the side effects of those antibiotics are worthwhile to study, because those would have beneficial research purposes. And then if you're giving antibiotics to treat the animals, what they're finding is that you're only getting about 1/10 of the dose that the animals need. So antibiotics in the water may be creating more problems like antibiotic resistance. So anybody have any idea what these are? Anyone? They're pinworms. So this is, I think these are Aspicularis. This has the lateral ailing. You probably remember this from boards, probably. So if you have these, you frequently treat with fenbendazole in the feed. So there was a group out of Johns Hopkins, and they treated with fenbendazole, and they actually treated some animals that had a brain tumor, and the brain tumor didn't grow. That's kind of interesting. So there was an article in the Baltimore Sun that says parasite treatment may help people with brain cancer. And so what they did is they looked at that whole category of drugs. So I think it was mebendazole was like the cheapest drug of that whole class. And what they do is it interferes with the microtubules with the tumor growth, and therefore prevented the tumor from growing. So this is one of those times when you get a research interference, which is a good thing, you know. Anesthesia, we talked about anesthesia. Anybody remember reading Curious George? Yep, was one of the first book I read twice in the library. I was so happy that I read that book. So ether can be a very dangerous thing. So this is the refrigerator where the ether was stored, and this is the door where the refrigerator was. So sometimes my environmental health and safety folks at some institutions said, oh, it's just flammable. That's kind of, it looks explosive to me. And there's multiple pictures of places like this. So be very careful, and that's why we really don't want to use ether if possible. Ketamine, xylazine. So here we find that in rats, if you're looking at parathyroid hormone or the consequences of parathyroid hormone, that could interfere with the research, especially if you're studying those type of things like calcium metabolism or many other things like that. So isoflurane. So if you have if you have an animal that might be getting, say, it's on a transplant study, like a kidney transplant or something to that effect, cyclosporin can increase the MAC for isoflurane. Different impacts on glucose levels and immune system problems that can develop from using isoflurane in some instances. Heating pads, we talked a little bit at our seminar. So this is an example of a burn that was seen. So you can see this is quite deep and quite severe. However, these can get so severe, there are reports out there of them causing renal injury. So please use water recirculating heating pads if you can, because you may end up causing renal problems potentially. So this last category is kind of crazy variables. I don't know what else to call them. Loco variables for the Spanish speakers out there. But things like earthquake. So the influence of various earthquakes that have happened. And this was a person who I think they got their PhD just studying how animals can predict earthquakes and what the impact of that is. So if you work in an area like California or Japan, even a place like Singapore, although there's no earthquakes per se, but we get a lot of the aftershocks from Indonesia, that could interfere with a lot of the activities and some of the behavior that you see with the animals, especially the behavior as before the earthquakes happen. Seasons are another one. So if you're in an institution that works with squirrel monkeys are very hardwired. So the summer months of, say, November to February in South America, they get what's called a fatted male syndrome. So the males will basically double their body weight. They'll get these very vibrant colors that they'll put on. So we get squirrel monkeys in the northern hemisphere, and they do exactly the same thing. They're on this November to February cycle where they get this fatted male syndrome. Same thing applies to some extent with mice. So mice in the winter depends on your local winter, but they're somehow able to pick up on the fact that it's winter. We get some breeding depression that we see. It's just amazing how some of these animals are hardwired. The one thing I really want to emphasize, and I have a whole host of slides that I received from Jerry Ward, it's really important as you develop your research project to consult with a veterinary pathologist. I think it's very helpful to look at what strain of mice you're looking at, the type of various models. I mean, there's a saying that veterinary pathologists know everything and can do everything, it's just too late. And I think there's an element of truth to that. So if you have the capability to reach out to a veterinary pathologist, I would highly recommend that. If not, there are several veterinary pathologists that offer consulting opportunities. And with the availability of really awesome technology like Aperio scanners, they can look at slides, they can consult like telemedicine in a way, and help with experimental design. I just can't say enough about that, and I found that to be very helpful from my researchers at several institutions. So that's the principal bit I want to talk about. I've taken on a new role since the beginning of the year working at Purdue, and we do have opportunities, and I wanted to just kind of highlight some of these for international visitors and for veterinary students. I talked at the veterinary school a couple of days ago. So we do permit visitors to kind of come in any time of the year. We'd like to have some clinical students, and there's a different variable duration that can occur of when the students are there. So those are great opportunities, educational opportunities. And they also extend into the faculty as well. I think they would like to have some faculty attend. And there's a lot of different opportunities here at the veterinary school. I mean, it's a big veterinary school, and they've recently gone through, I think about 3 years ago, they basically have a whole new veterinary school. So the whole layout and everything is quite quite interesting and modern. I'm still trying to wrap my head around it because I went to the old vet school back last millennia. So I think it's a great opportunity. And just want to thank everyone for everyone here has been so wonderful over the past several days, generous hospitality. And I just want to thank everyone for the opportunity to come back again this year and talk to everyone. Thank you. Have any questions? Yeah.
Do you have any information on microbiome, gut microbiome, exactly research?
Yeah, I have some slides on that, and I think that's it's interesting to bring that up because even we started doing that when I was at Perth, and when I talked to Jax, I said, "Why do you want to do that?" And I said, "Well, we look at the health of the animals, and we publish that online." So our goal, and I don't think it all got happened for everything, but I know at least for the Black 6, because that's the most commonly used, that we publish that data because there were people starting to look at microbiome. I mean, we started that, and then COVID hit, and then no one cared about the microbiome. It was just all about COVID. So I think that in the future, we're going to see microbiome data that's going to be available. But if I take a cage of mice in a room, determine what their microbiome, if I move them to another room, same food, same water, same care technician, for about 2 weeks, their microbiome is all messed up. So it's a big deal. We all experience that when we travel. You know, the first thing I hit is I like the curd because it maybe helps my microbiome a little bit. But I think that's something we're going to see more of because I think sometimes when research doesn't work, it's always the veterinarian's fault why the research doesn't work. But I think sometimes it's just the microbiome. We're putting the animals in there. Even things like fenbendazole feed or ivermectin, I mean, those are antibiotics. Okay. Let's see here. I'll try to, yeah.
Thank you very much, Dr. Patrick Sharp, for your insightful presentation. We would like to invite Dr. Varuni Gunatilaka to present a token of appreciation to Dr. Patrick Sharp. Once again, thank you very much, Dr. Patrick Sharp and Dr. Gunatilaka. Ladies and gentlemen, it's now the time for the second guest speech of today by a distinguished figure whose commitment to laboratory animal research in Sri Lanka has been truly remarkable. We are honored to welcome Professor Mangala Gunatilaka, Professor at the Department of Physiology, Faculty of Medicine, University of Colombo, Sri Lanka. Professor Gunatilaka, as the founding president of Sri Lanka Association for Laboratory Animal Science and the president for the term 2023-2024, has dedicated her professional life to advancing laboratory animal science in Sri Lanka. Her leadership and tireless efforts have played a crucial role in shaping the laboratory animal science landscape in Sri Lanka. Today, Professor Gunatilaka will speak on 12 years and beyond commitment to laboratory animal research in Sri Lanka. To introduce this esteemed speaker, I now invite Dr. Ureshani Karunaratne to deliver the introduction. Over to you, madam.
Thank you. So I would like to invite our next speaker, Professor Mangala Gunatilaka. Vidyanidhi Professor Mangala Gunatilaka is a distinguished and influential figure in the field of laboratory animal science, affiliated to the Department of Physiology at the Faculty of Medicine, University of Colombo. She made the history in 2011 by establishing the first international certificate course in laboratory animal science in the Asian region in collaboration with the Utrecht University, the Netherlands, thereby pioneering in laboratory animal science education in Sri Lanka and setting a standard for future programs. As a founding president of the Sri Lanka Association for the Laboratory Animal Science, her leadership has been pivotal in advancing the field within the country. She introduced innovative alternative research models such as zebrafish embryo model, which have had a transformative effect on the research practice in Sri Lanka. Under her guidance, SLALAS has thrived and she completed the second term as a president in 2023 and 2024, during which she continued the introduce groundbreaking concept in enhance the field. Beyond her role with SLALAS, she also the founding president for the Society for Alternative to Animal Testing in Sri Lanka and is the founding director of the 3R Center for LAS, emphasizing the principle of replacement, reduction, and refinement in the research methodologies. Professor Gunathilaka's contribution have not gone unnoticed. She has earned numerous accolades on both national and international stages, highlighting her significant impact in recognition as a leader in laboratory animal science globally. Her work continues to inspire others in the field and fosters more ethical and scientifically sound approach to research practices in Sri Lanka and beyond. Over to you, madam.
Good morning, all of you, our distinguished guests, executive committee members, and dear students. And first of all, I would like to thank Dr. Ureshini Karunaratne for your generous introduction about me. And also I would like to thank you. So this topic about the 12 years and beyond, topic was designed by Dr. Sachini, and she invited me to concentrate on this SLALAS journey. And during this introductory speech that is related to our journey, I am going to concentrate about the SLALAS because I would like to mention several things about SLALAS, especially because of the presence of our students. And in the second section, I am going to concentrate a little bit about the One Health concept because in the third section that I am going to bridge in the SLALAS with the One Health concept. So these are the three things that I am going to concentrate on. And the first two sections, those are very tiny sections. The main section is the third one. So for the students, I would like to mention that SLALAS was established in 2012, that is on 21st of December. So we have completed 12 years by 2024 December. And our mission is to, the SLALAS is dedicated to enhancing animal welfare and promoting the ethical use of laboratory animals by applying the 3Rs principle in laboratory animal science. That is our mission. And in our constitution, there are several objectives with different verbs used in the objectives. And to advance, to promote, to encourage, to cooperate, again to promote, and to be the monitoring body, that is the last one. That is about the ethical conduct of laboratory animal experiments in the country. I'm not going to read all the objectives. If you are interested, especially students, so you all can go to the website of the association, and all the details are given there. If I concentrate on the second part, yesterday during the presidential speech, Dr. Sachini briefly mentioned about the One Health concept. But in order to complete this guest speech, I would like to again concentrate on this One Health concept. It is a concept that has been known for more than a century, that human, animal, and plant health are interdependent. And also, it is not only interdependent, that is interconnected and bound to the health of the ecosystems in which they exist. We envisage and implement it as a collaborative. It is not individual item, it is a collaborative. Whole of society, whole of government approach to understanding, anticipating, and addressing risks of global health. So this is the information, introductory information given in the website, the World Organization for Animal Health. If I concentrate on the WHO information, it mentions that it is an integrated, unifying approach to balance and optimize the health of people, animals, and environment. Again, I am emphasizing the interconnectedness in these three things. Why this concept is important? It is to prevent, predict, detect, and respond to global health threats such as the COVID-19 pandemic. Some other important facts. So, there is a need for multiple sectors, disciplines, and community involvement, interconnectedness. So, all should be work together, not in different compartments. Development of new and better ideas to address root causes and create long-term sustainable solutions. One Health approach is particularly relevant for food and water safety, nutrition, control of zoonotic diseases, pollution management, and combating antimicrobial resistance. So, as laboratory animal scientists, we know that we are involved in research related to all these sections. I am going to bridge the two things, SLALAS with the One Health concept. What are our contributions during this 12-year journey? Over the past 12 years, SLALAS has contributed to advancing this multidisciplinary approach through various initiatives and activities. SLALAS has made an impact through, so there are sub-areas, education and training, capacity building, research collaborations, guidelines development, international collaborations, biodiversity conservation, and finally about public awareness campaigns. SLALAS has focused on enhancing knowledge and skills related to laboratory animal science, particularly in the context of One Health. That is by organizing courses, workshops, seminars, webinars, training sessions, various competitions, and through publishing newsletters and newspaper articles. The association has educated researchers and students. and the general public about the importance of ethical practices in the use of laboratory animals and their significance in medical research. So we have started this education and training in 2013. So we established in 2012 and we commenced our education and training programs in 2013. So you can see the first workshop that we conducted that was organized by Professor Sugandhika in her university. And this one, Dr. Prasadi, when she was at Uvavellas, she organized this one. So we didn't want to conduct all the things in Colombo, but we visited other institutions or universities. And one of the important agenda item in our laboratory animal education, it is the course that we conduct. So these 3 flyers you can see, I didn't include all the things throughout, that is starting from 2013, but this one about the 2023 one, 22, 2023 and 2024 flyers that we prepared. And this is the last course that we conducted, that is last year, 2024. So practical sessions were almost every year. There were exceptions, few exceptions, but almost every year we conducted these training sessions in the Medical Research Institute Animal Center. So following education and training, I would like to concentrate on the second sub-area, that is about capacity building. By enhancing the capacity of local researchers, students and supportive staff, SLALAS has promoted sustainable practices in laboratory animal science. Developing local expertise ensures that research conducted in Sri Lanka is responsible, ethical, and aligned with international standards. This was conducted, that is in December, again organized by Dr. Prasadi at the University of Colombo on advanced workshop on zebrafish as a research model. And we also conducted several other workshops and discussions, that is online discussions. So this is about, the first flyer is about the C. elegans workshop that was organized last year. And the other one, it is again about the ethics and ethics of research involving wild animals and lab animals that was conducted again last year. And again, even in the last year, we didn't, that means restricted our education and training programs to Colombo. So we were able to obtain a grant from Laboratory Animals Limited UK, and we were able to go to the eastern part of the country and also the northern part of the country. So in the Jaffna University and also in the Batticaloa University of Sri Lanka. That is about ethics and culture of care. This is the Jaffna University session that we conducted about the lab animal ethics and culture of care. And also this was organized in collaboration with AFLAS. So we obtained AFLAS approval, that is to use their logo in this one. And also this was conducted as a pre-activity commemorating the contribution of lab animals for the medical sciences, for the progress of medical sciences. And this one that is about application of cell cultures and make compliance, real-time PCR assay. That was again conducted with University of Peradeniya and also in collaboration with Sri Lanka College of Veterinary Surgeons, but this one organized by our association. Two other activities, again one on cell culture and the responsibilities and challenges. That was an important session that we conducted in 2022 during Dr. Kalpani Ratnayaka's tenure of office as the president. And we didn't limit some of the areas that we covered because we conducted a pre-conference workshop on microbiological monitoring and genetic monitoring of laboratory animals. That was in 2024 January as a pre-conference workshop, and also a pre-conference workshop on systematic review and meta-analysis. So we didn't limit our scope to certain areas, but we address some other outside areas as well. So Dr. Patrick Sharp and Professor Cholawat, so your involvement in the pre-conference sessions that we concluded very recently. So rodent anesthesia and analgesia and exploring analgesia and green practices in animal vivariums. So these are organized by the association in collaboration with the Medical Research Institute and the College of Veterinary Surgeons. Some other areas that we concentrated on last year, that is about the project management, which is important for the information that is for our students. And also, we wanted to concentrate on the current areas. That is, the topic that was addressed, will artificial intelligence finally put an end to animal testing? That was conducted in collaboration with the Society for Alternatives to Animal Testing. So we thought that it is not only the researchers, research students, that we wanted to have some education planned for animal house technicians and other staff. So because of that, we conducted this culture of care workshop that was in 2023. And this was conducted at the Faculty of Medicine, University of Colombo, and the practical session was conducted at the Medical Research Institute Animal House. And about research collaborations. Through the members, association has fostered collaborations among researchers from different disciplines, promoting interdisciplinary research that addresses health issues at the human-animal environment interface. This collaborative research has supported the development of new therapies, innovations, and diagnostic tools that benefit both human and animal populations. And the examples that I selected for this presentation, the first one, that is Dr. Ratna Divakara's PhD project one area, detection of immunity in sheep following anti-rabies vaccination. So part of the analysis was conducted in the Department of Physiology, Faculty of Medicine, and we were able to establish the ELISA technique in the department for this purpose. And also another area that we concentrated on, that is following the guidance of Dr. Suresh Pusala, the capability maturity model as a measure of culture of care in laboratory animal science. And another publication, as you can see, evaluation of antipyretic effect of Sudarshana syrup on vista rats. So the experimental or research were carried out using vista rats, and the research team were able to develop the Sudarshana paneer for the benefit of especially the pediatric population. Not only capacity building, not only promoting research, not only education and training. So this offering seed grants by the association was initiated last year. So we advertised, applications were called, and there were 3 applications received and 2 were selected, and it was announced yesterday. We contributed for the guidelines development as well, advocating for policies that support laboratory animal welfare and ethical research practices has been a crucial part of Sri Lanka Association Laboratory Animal Sciences mission. By influencing different sectors of stakeholders and the association has contributed to the development of guidelines that ensure the humane treatment of animals in research, aligning with One Health principles. We are in the process of influencing the policymakers. About international collaborations. Through partnerships with global organizations, ICLAS, SLALAS, etc., and participation in international conferences and organization of international conferences like this, SLALAS has shared knowledge and best practices related to laboratory animal science and One Health. These collaborations have helped position Sri Lanka as an active participant in the global health-related discussions. So myself and Dr. Sachini got an opportunity to participate in the SLALAS conference held last year. It was the 17th SLALAS conference, and we were actively participated in discussions related to SLALAS Congress organization that will be held in China this year, this October, and also about the journal that is published by the China Animal Models and Experimental Medicine Journal. And because of the international collaborations, so we got an opportunity to select our members, young members, to participate in SLALAS conference. So for the very first one, Dr. Kalpani was able to participate, and this is the last one, that is last year. Dr. Anusha was able to participate in this. So it's a great initiative by the SLALAS. And these are the flyers related to the international conferences that we conducted in 2023, 24, and now in 2025. So all these with the participation of international members. So this is some of the snapshots that were taken at the last conference, that is in 2024. So you can see Professor Vera Bowmans, Professor Patricia Turner, and Professor Cynthia Peko, former ICLAS president, and Dr. Suresh, you are there. Dr. Ingle is there, and Professor Cholawat and Dr. Patrick. So with the participation of all these international people. And about biodiversity conservation, that is SLALAS contribution towards this effect, one of the One Health sections. Recognizing the importance of biodiversity to overall health, SLALAS has been involved in initiatives that promote the conservation of the ecosystem. The association contributes to a broader understanding of health risks and environmental sustainability. So this is the one that we planned for the Asia Laboratory Animal Day 2024. So Asia Laboratory Animal Day was declared as 29th November. That was initiated last year, that is in 2024. So Dr. Sachini, under her leadership, she organized this tree planting program with the participation of the Vice Chancellor of University of Colombo. And you can see in the second picture some of the academic staff members of University of Colombo and the executive committee members who participated in this event. So we didn't limit the activity, that is for the commemoration of the contribution made by the laboratory animals. We had another session that is in my department, the Faculty of Medicine, University of Colombo. So SLALAS, in collaboration with the Triage Center, inaugurated a laboratory animal science corner at the Faculty of Medicine, University of Colombo. The Laboratory Animal Science Corner is a unique initiative that houses a museum and a book collection, both of which serve to highlight the significant history and contributions to laboratory animal science in Sri Lanka. This Laboratory Animal Science Corner will serve as a valuable educational resource for students, faculty, and researchers, while also commemorating the rich history and ongoing advancements in laboratory animal science within Sri Lanka. Finally, that is about the public awareness campaigns. From the inception, SLALAS has engaged in public awareness initiatives aimed at informing about the importance of One Health. By providing information on the role of laboratory animals in advancing medical sciences, the association has helped to foster a greater understanding of how interconnected these various health domains are. So you can see the paper cut that was published in Sunday Times. So this was written by the deputy editor, Mrs. Kumuduni Hettiarachchi. That is, so she participated in our inaugural conference held in 2014. So she wrote about this one, so to create public awareness about our activities. It was not only limited to newspaper publications. SLALAS has engaged in public awareness initiatives which are aimed at educating the student community about the importance of One Health by organizing various competitions. So art competitions, oratorical competitions, photography competitions, and poster competitions. And we have planned a laboratory animal scientist and media roundtable discussion today. So if the media personnel will be here for that one, we will conduct that one. So these are the objectives of that session. Finally, SLALAS has significantly contributed, as you all have seen, to advancing the One Health approach over the past 12 years. Through education, capacity building, research collaboration, public awareness, guidelines development, biodiversity conservation, and international collaboration, SLALAS has played a vital role in promoting the holistic understanding of health that encompasses human, animal, and environmental well-being. So in our capacity, we contributed in various ways to achieve these subsections related to One Health. As One Health continues to gain importance in addressing global health challenges, SLALAS' contribution will prove invaluable in fostering a healthier world despite the challenges we face. With that, I would like to end my presentation and thank you for your patient listening. So this is a kind of message, especially to our students. So you all have to concentrate on these aspects when you are going to conduct different research activities. Thank you.
Thank you very much, Professor Mangala Gunatilaka, for your inspiring and wonderful presentation, a chance for us to go back and revisit the journey that SLALAS has come through the years. And now I would like to invite once again Dr. Reshani Karnaratna to present a token of appreciation to Professor Gunatilaka in recognition of her outstanding contributions. Once again, thank you very much, Professor Mangala Gunatilaka, and thank you very much, Dr. Reshani Karnaratna. With that, we will now take a short break for the morning tea, a perfect opportunity for you all to refresh and connect with the fellow participants. And please be sure to return back to the hall within 15 minutes of your break. See you once again. Hi everyone, welcome back to the 12th annual session and international conference of Sri Lanka Association for Laboratory Animal Science, and we hope you had a nice break. After the break, the first thing that we are going to do is ask a question from you all. So the enthusiastic participants and our students, this is your chance to win a prize based on the sessions that we had today. So the question is, we've been talking about crazy variables today. Mention 2 crazy variables that can affect animal experiment. Okay, now there's a competition. Who's the one who raised the hand? Yes, I think it's ladies first. Okay. Well, that's the correct answer. Okay. Earthquakes and seasonal changes. We invite Dr. Patrick Sharp to give the gift to the winner, and winner, come on stage to receive your gift. Congratulations to winner, and thank you very much, Dr. Patrick. With that, we are privileged to welcome Dr. Satahiro Nakio from the Center for Animal Resources and Development from the Division of Reproductive Engineering, Kumamoto University, Japan. Dr. Nakio will present on emerging technologies for regulating reproductive performance in rodents. I would like to call upon Dr. Kalpani Ratnayaka to introduce Dr. Satahiro Nakio. Over to you, madam.
Thank you. It's my pleasure to introduce Professor Satahiro Nakao. Professor Satahiro Nakao is a program assistant professor at the Division of Reproductive Engineering, Center for Animal Resource and Development, Kumamoto University, Japan. With expertise in reproductive biology and advanced reproductive technologies, Dr. Nakao has significantly contributed to improving fertilization methodologies and gamete biology. Professor Nakao completed his PhD in medicine from Kumamoto University in 2021, building on a strong academic foundations with a master's and bachelor's degree in medical and pharmaceutical sciences, respectively. His research focuses on optimizing in vitro fertilization and sperm cryopreservation protocols with published work in high-impact journals such as biology of reproduction and scientific report. Prior to his current role, Professor Nakawo was a research fellow at CARD and technical staff at Niigata University's Brain Research Institute. His innovative research has earned him accolades like the Research Encouragement Award from the Society of Animal Reproductive Biotechnology and the Best Poster Presentation Award from the Pharmaceutical Society of Japan. A member of the International Communication Committees of the Japanese Association for Laboratory Animal Science, Professor Nakawo is dedicated to advancing international collaboration in reproductive biotechnology and laboratory animal science. It's our pleasure that you accepting our invitation and with us today, and I am cordially invite you to deliver your talk. Over to you, sir.
Hi, I'm Satohiro Nakao, and first, I'm so grateful to have the opportunity to talk about my research here. And now I'm still studying English now, so I'm concerned about my English skills, that I'll do my best. First, I'd like to introduce myself. I'm Satohiro Nakao. I'm working as a program assistant professor at the Center for Animal Resources and Development, CARD, at Kumamoto University, Japan. And since last year, I have been a member of International Communication Committee of Japanese Association for Laboratory Animal Science. And my major is pharmaceutical science, medical science, and medicine, and I have received research instructions from Professor Naomi Nakagata and Professor Toru Takeo. And I like cycling and playing the music, and recently I started shakuhachi, which is a Japanese traditional flute made from bamboo. And I met Dr. Sachini at the AFRAS conference and Young Scientist Camp in 2023. And this encounter has given me this great opportunity. So thanks, Sachini, to giving me this opportunity. And now I'm working on developing the development of reproductive technology for mice and rats. So in this presentation, I'd like to introduce emerging technologies for regulating reproductive performance in mice and rats. CARD is a research center for genetics and biomedical science using genetically engineered mice. And CARD is also a quality center of mouse resources as a mouse bank. And we also, in 2019, we also started rat banking services. The CARD provides the services for the collection, production, cryopreservation, and supply of genetically modified animals. And in our laboratory, we continuously develop reproductive technologies to improve the efficient banking system of mice and rats. So today, in this presentation, I'd like to introduce our recent advancement of CARD reproductive technologies for mice and rats. And all experiments are approved by IACUC. And today, I have total 6 topics, and 4 topics are for the mouse reproductive engineering techniques, and 2 are for the rat reproductive technologies. The first topic is mouse sperm selection. One of the major reproductive engineering techniques is in vitro fertilization, IVF. IVF is a technique where sperm and eggs are fertilized in vitro. mammalian sperm don't have fertilization ability immediately after collection. We need to induce capacitation. Capacitation is sperm activation, and sperm can be fertilized after capacitation have occurred. So, sperm capacitation is an important factor to have a successful IVF. Therefore, to enhance the fertilization rate, use only capacitated sperm for IVF or induce capacitation with high efficiency might be effective. So, to improve the IVF techniques, we tried developing the mouse sperm selection technique. First, we tried collecting the mouse sperm by using a general cell sorter, but we could obtain a small number of sperm, and that sperm didn't move forward. The sperm tail was just only slightly moving. So, to collect the sperm with motility, we need to choose a special cell sorter. So, we used the microfluidic strip cell sorter. This cell sorter uses air pulse pressure to separate the cells, resulting in minimum damage to cell. So we applied this cell sorter for mouse sperm collection. By using the microfluidic strip cell sorter, we could obtain sperm with motility like this video. Next, we actually tried selecting the mouse sperm with high fertility. We use acrosome reaction as a capacitation marker, and we detected it using PNL lectin FITC staining. We separate the cell PNL low group and PNL high group. After selection, these sperm showed a good motility. And then we actually performed the IVF, and PNL high sperm showed a higher fertilization rate. So we could have success in selecting the mouse sperm with high fertility. And furthermore, we confirmed the developmental ability of the embryo derived from selected sperm. The embryos derived from PNL low and high sperm normally developed into blastocyst via in vitro culture, and after embryo transfer, the embryos were normally developed into live pups. And this is a topic summary. By using a microfluidic strip cell sorter, we could have a successful selection of mouse sperm with high fertility based on acrosome reaction. So we think these findings would be useful for improvement of IVF techniques and development of new ARTs. The second topic is new capacitation inducer. So to enhance the fertilization rate, inducing the capacitation with high efficiency also might be effective. So we tried searching a new capacitation inducer. And now, cholesterol efflux is thought to be the trigger of capacitation. And actually, cholesterol acceptors such as albumin and methyl beta cyclodextrin, LBCD, are used for sperm capacitation inducer. Treatment of BSA and LBCD enhance the cholesterol efflux from the sperm membrane and increase the fertilization rate. And recently, we found a new capacitation inducer dimethyl alpha cyclodextrin, DMCD. DMCD enhances sperm fertility, and very interestingly, DMCD did not enhance the cholesterol efflux, but enhance the phospholipid efflux. So this result suggests that there is a new mechanism of capacitation that doesn't involve cholesterol efflux. And in addition, we confirmed the developmental ability of the embryos treated, derived from DMSCD treated sperm. These embryos were normally developed into blood cysts and normally developed into live pups. And we also confirm whether the DMSCD can normally induce sperm capacitation. We evaluated the several capacitation markers. First, we evaluated the membrane fluidity. It is known that the membrane fluidity is increased in capacitated sperm. As with BSA and MVCD, DMSCD also increased the membrane fluidity. And next, we evaluated the lipid raft localization. It is known that the localization of lipid raft is changed with capacitation, and upper pattern was changed to lower pattern after capacitation. As with BSA and MVCD, DMSCD induced lipid raft relocalization. And finally, we evaluated the acrosome reaction. Acrosome-reacted sperm can be fertilized with oocyte. As with BSA and MVCD, DMSCD also induced the acrosome reaction. This is a topic summary, and we found that DMSCD enhanced sperm fertility by phospholipid efflux. The general mechanism of capacitation is a cholesterol efflux, but we found a new mechanism of capacitation, and the phospholipid efflux would be a novel trigger of capacitation. And these findings would be useful for improvement of IVF techniques and development of new ARTs. And fortunately, this research was featured in newspaper and on TV in Japan. And my wife, after watching TV, my wife said, "You really were a researcher." So finally, I was recognized by her as a researcher. So this may be the biggest achievement for me. Okay, so the third topic is synchronized mating with the ovulation timing. Mate is another way to obtain fertilized oocytes, and by using mate and embryo flushing, we can obtain in vivo fertilized oocytes. In general mating, a female at estrous stage or a female-induced superovulation is cohoused with male overnight, and next morning we can obtain fertilized oocytes. However, the problem in conventional mating is that the limited number of oocytes that can be fertilized in the oviduct, resulting in a low number of fertilized oocytes. For example, we use 4 weeks old female mice, and we use IAC for superovulation. There are many oocytes in the oviduct, but we can obtain only around 20 fertilized oocytes. So to overcome this problem, we examine the effect. Of timings of ovulation and mating. First, we check the ovulation timing after HCG injection, and based on this data, we define the ovulation timing 3 periods: pre-ovulation, during ovulation, and post-ovulation. And then we performed mating at these 3 periods. The successful mating rate was high at all these 3 periods, But mating at during and post-operation improve the fertilization rate and increase the fertilized oocytes. So we could have success in improving the fertilization efficacy in vivo. And the embryos obtain the synchronized mating normally developed into blastocysts via in vitro culture and normally developed into live birth after embryo transfer. And furthermore, we confirm when we can obtain the fertilized oocyte at pronucleus stage. The in vivo fertilized oocytes are often used for genome editing, and for genome editing, the pronucleus stage fertilized oocytes are useful. When we perform the post-ovulatory mating 9 a.m. to 1 p.m., we could obtain the pronucleus stage fertilized oocyte from 5 p.m. to 7 p.m. So these mating techniques can be applied to produce the in vivo fertilized oocyte for using for genome editing. This is a topic summary. The synchronizing the timings of ovulation and mating increase 3 times higher number of fertilized oocytes. And this technique is useful for efficient production of in vivo fertilized oocytes and contribute to 3 hours, reducing the number of oocyte donors. The last topic for mice is suppression of the sperm fertility. This is our latest study published in Biology of Reproduction last month. As you know, the sperm oxidation is one of the major causes decrease the fertility. And in the study, we focused on the cell oxidation, and we used DTNB, which is a membrane impairment cell selective oxidizing agent, and we confirmed the fertilization ability. DTNB treatment oxidizes the cell groups in the sperm membrane and decreases the fertilization ability of sperm. Next, we evaluated the sperm motility parameters, motile and ALH. ALH is an indicator of sperm hyperactivation, and hyperactivation is change in motile necessary for sperm to fertilize. The DTNB did not affect the motile but decreased the ALH. And furthermore, we evaluated the intracellular calcium ion level. The influx of calcium ion is important for sperm hyperactivation. DTNB treatment decrease the intracellular calcium ion levels in sperm. This is a topic summary. The oxidation of cell groups in sperm membrane protein can decrease the fertility of sperm by suppressing the calcium ion influx and hyperactivation. This finding provides a new insight into the role of cell groups in sperm membrane. Okay, so last 2 topics, I'd like to introduce the rat reproductive engineering techniques. The first topic is rat sperm cryopreservation and in vitro fertilization. Laboratory rats are the most used experimental animals after mice. Rats have many benefits compared to mice. For example, they have a larger body size and they have a higher cognitive function. And recently, we may produce useful disease model rats. So the usefulness of rats as experimental animals is attracting attention. However, the infrastructure for reproductive engineering techniques in rats is not established satisfactorily compared to that of mice. For example, in cryopreservation, The rat sperm is so sensitive to physical stress because the rat sperm is longer than the other animal sperm. So cryopreservation of rat sperm was so difficult. But recently, we could have success in developing the optimized protocols for rat sperm cryopreservation and IVF. First, we found that cooling before sperm cryopreservation was effective in avoiding the cryo injury. Without pre-cooling, the sperm is actively moving in the cryopreservation medium. But with cooling, we can inhibit the sperm moving in the cryopreservation medium, and that is important for sperm to maintain high motility after cryopreservation. And we also work on improving the IVF using cryopreserved rat sperm. And we found that a high concentration of VSA improves the fertility of cryopreserved rat sperm. And also, the removing the tumoral cells surrounding the oocyte also improves the fertilization rate of cryopreserved rat sperm. This is a topic summary, and pre-cooling is important to maintain high motility after cryopreservation, and high concentration of VSA and removal of tumoral cells were effective in IVF using cryopreserved rat sperm. These techniques would be useful for efficient preservation and production of rat resources and promotes the research using genetically modified rats. The last topic is a cold storage of rat sperm and IVF. The transport technology of laboratory rats is an important topic in reproductive engineering techniques. In general, the transport of live animals is used to share the rat resources. But transportation of live animals has many disadvantages, such as the risk of death and escape, and we need to consider the animal welfare, and we need a high transportation cost. The cold transport of sperm provides a simple solution for sharing the rat resources, but the decrease in fertility with prolonged storage periods is a problem. And recently, we found that the DMSO and quercetin and high concentration of BSA are effective in solving this issue. And our master's student, Keisuke Masuda, is having a poster presentation about this topic, so I will only briefly introduce the data here. So adding the DMSO and quercetin increase the fertilization rate of cold stored rat sperm, and using a high concentration of VSA increase the fertilization ability, and we could store rat sperm at refrigerated temperature for up to 6 days. So please visit Keisuke's poster presentation and ask him the detailed information. This is a summary. So those are the our emerging technologies for regulating reproductive performance in mice and rats. By using these techniques, we can efficiently produce the mouse and rat fertilized oocytes and contribute to the reduction of animal numbers following the 3 R's. And I would like to continue establishing the research support system through the research and development of reproductive engineering techniques and contribute to life science, medical science, and pharmaceutical science. This slide lists my collaborators, and that's all. Thank you for your kind attention.
Thank you very much, Dr. Satahiro Nakayo, for your insightful presentation. We would now like to invite Dr. Kalpani Ratnayaka to present a token of appreciation to Dr. Nakayo. Once again, thank you very much, Dr. Satahiro Nakayo. With that, we are moving on to one more questions to our participants. So the question is, when was the Sri Lanka Association for Laboratory Animal Science, shortly known as SLALAS, being established? Okay, the lady in blue. Do we take that as the correct answer, Professor Mangala? Okay. Yes, it's the correct answer, and we invite Professor Mangala Gunathilaka to give away the gift for our winner. Thank you very much, Professor Gunathilaka. And there's one more chance for these exciting participants who are willing to win one more gift. So the question will be, state the difference between mice and rat sperm cryopreservation techniques. I'm going to repeat the question. State the difference between mice and rat sperm cryopreservation techniques. Shall I repeat the question one more time? Okay. They're searching. Right. So we are going to nullify the question because the time taken to answer the question was little bit long than the questions that we've asked before. So we are going to nullify the question, but we'll be having more questions in our session coming up. So you have still chances to win a gift from us. So stay tuned and listen to the lectures attentively. With that, ladies and gentlemen, we now move on to a highly anticipated moment, the announcement of the photography competition winners. This competition has highlighted the creativity and talent of our participants, and we are extremely excited to recognize their outstanding contributions. To present the awards, I'd like to invite Dr. Vindhya Fernando, accompanied by the President of Sri Lanka Association for Laboratory Animal Science, Dr. Sachini Amarasekara, to join us on stage. Now it's my pleasure to announce the winners of the photography competition conducted by SLALAS. The second place will be shared between A.M. Nilushika Dhananjani Senaviratna and Yadugasheni Srikunageshwaran. And the first place goes to A.M. Nilushika Dhananjani Senaviratna. Please join me with a huge round of applause in congratulating these talented individuals for their remarkable work, even though they are not here today with us. And now it's my pleasure to introduce Dr. Arvind Ingle, the President of Indian College of Laboratory Animal Medicine. And Dr. Ingle will speak on infectious diseases of laboratory animals today. I now invite Dr. Hasanthi Ratnadivakara to introduce Dr. Arvind Ingle.
It's with great honor I introduce Dr. Arvind Ingle to this audience. Dr. Ingle is a renowned veterinary pathologist and a prominent leader in laboratory animal science with over 3 decades of expertise in the field. Currently, he serves as the governing board member and chairman of the Education and Training Committee of the International Council for Laboratory Animal Science, ICLAS, driving global advancement in research and education. Dr. Ingle obtained his MVSc in veterinary pathology from the Postgraduate Institute, Akola, India in 1992 and earned his PhD in 2012. His groundbreaking research spans animal model development, microbiological and genetic quality control, embryo cryopreservation, human tumor xenografts, and comparative pathology. He made a landmark contribution in 2010 by reporting the world's first laboratory mouse papilloma virus in veterinary pathology. A fellow of both Indian Association of Veterinary Pathologists and the Laboratory Animal Scientists Association, Dr. Ingle recognized for his significant contribution to the field. In 2024, he also earned distinction as a GLP inspector for the NGCMA Government of India. Dr. Ingle has published over 110 peer-reviewed articles, garnering 2,550 citations and an H-index of 30, and has delivered more than 160 invited lectures at national and international forums. He serves on the editorial boards of 15 prestigious journals and has chaired the Board of Studies in Veterinary Pathology at MAFSU Nagpur. Among his many honors, Dr. Ingle received the Best Laboratory Animal Pathologist Award 2013 and the Turnkey Facility Leader of the Year Award from ALN Magazine USA in 2014. And also, Dr. Ingle have been nominated by the Government of India as a Central Committee member of the Committee for Control and Supervision of Experiments on Animals. A past president of both Laboratory Animal Scientists Association in India and the Asian Federation of Laboratory Animal Science Associations, Dr. Ingle continues to shape global standards and education in the laboratory animal science. With that introduction, I would like to invite Dr. Arvind Ingle for his guest speech.
Good morning, everyone. At the outset, I take this opportunity to thank the SLALAS for inviting me to share my experience on the topic infectious diseases of laboratory rodents and their diagnostic importance. So I chose this topic as the uh, theme of the conference is towards One Health, responsible and reproducible animal-based research. So unless we use the quality animals, we cannot achieve the reproducible results. And in my presentation, I am going to explain how the infection may interfere the research, and that is what the topic is important for us. The background of the talk would be, as we know, the laboratory animal are inevitable in laboratory research, and like us, the laboratory animals are susceptible to a variety of microbes, and some of them are lethal. Some they are clinically, they show the clinical symptoms with high or low morbidity and mortality. Majority of them are asymptomatic, and but both of them, the clinically symptomatic and the asymptomatic, they interfere with the research of all kinds, and there lies the diagnostic importance of, or what we call the quality control. So as we know, what are the animal species used are mammals, reptiles, birds, as well as aquatic, and all of them are prone to the infections. So as we know, mice, rat, which constitute more than 90% of the junk for the animal research. Similar way, hamster, rabbit, guinea pig, and aquatic animals. There are other animals, but my talk would be restricted to these 4 or 5 species. So for the consistency, what we call the uniformity and the reproducibility, these are the factors responsible. What kind of animal breeding we use for producing the animals, what are the experimental protocol, whether it is foolproof, expertise of the principal investigators, what are the food and water we provide to the animals, controlled environment, experienced technical staff, and more importantly, the quality of animals. So these are the variables beside the food and other practices. These are the major variables wherein when the PI don't get the required research output, we must think of the microbial status, bacterial, viral, and parasitic load of the animal. Genetic, what are the sources from where we procure the animal, and whether they are contaminated genetically or if they have any mutation. And as we know, the light, temperature, humidity, and these are the factors, environmental factors. My talk would be restricted to this, although we also have the genetic monitoring program at my place, and we are the only in India which does the genetic monitoring and also provide the services to outside institutes if you visit our website. There are metabolic diseases also, but we are restricting to microbial infections. So what consequences the microbial contamination cause? They alter the results of the research. They reduce reproducibility, they render the model unuseful, they contaminate the biological material which we use for the experimentation, like tumor cell lines, normal cell lines, sera, bacteria, viruses, and parasites, if we are working on these for the experimentation. These are the research complications when we use the contaminated animals, and we call them artifacts, and they are rarely published, and if at all scattered, and we are not aware of that. So it is our duty that the artifacts are also to be published, so that the researchers, they know what are the research complications through these infections. NIH guide has characterized the microbes as per the category A, B, C, D, E. They are zoonotic, pathogenic to fatal to animal, pathogen not fatal but can cause disease in animals, opportunistic, and indicator. A whole list of viruses, bacteria, as well as parasites are given in the NIH handbook, which is freely available in Google. Whereas in Pelas also, there is a lot of least battery of bacteria, viruses, and additional agents also for different species, including hamster, rabbits, guinea pigs, and I will be restricting to some of the important viruses and bacteria as the time is restricted. So hantavirus in rodents is one of the important virus, and this is health hazard to person working in the animal facility. This has zoonotic importance, and it is a life-threatening hemorrhagic disease in humans, and it affects brain, liver, lungs, and spleen lesions. And in India also there was in 2011 there was a report, and the cause was because of the rodents. It was published in the news in 2011. This is how the interstitial inflammation as well as the edema in lungs is the finding and the cause in case of Hanta. Lymphocytic choriomeningitis is again zoonotic disease. It causes glomerulonephritis. It influences humoral and cellular immune response, causes a long-standing immunodepression and transient bone marrow aplasia. It enhances the frequency of lymphoma after treatment. So if we are working on lymphoma and the animals are infected with LCMV, they are no good model for the research for the uh, lymphoma studies. This is how the normal brain in case of children, and you can see the hypoplasia and also the gyra and sulci results into changes. We diagnose LCME by ELISA as well as PCR. Next virus is Sendai virus. It increases mitotic cells in bronchi and lungs. Edema increases bronchial mast cells. So if someone is working on lungs for the tumor development, because of the mitotic cells, there is a misinterpretation of the results of the research, because the Sendai viruses also produce the mitotic cells. It reduces tumorigenicity and transplantability. So the tumor, mouse to mouse tumor transplantation is also hampered. It increases NK cell cytotoxicity and delayed wound healing. So someone working on the wound healing and the animals are infected with Sendai virus, they are no good model for the research. This is how you can see disorganized bronchial and metaplasia and hyperplasia, as well as giant cells in the lungs when the animals are infected with the Sendai virus. This can be very well diagnosed. At our place, we use the PCR to diagnose these viruses. Murine norovirus, this is the recently imported virus when we import the animals. There is no disease in immunocompetent mice. MNV has a tropism to infect macrophages and dendritic cells. It infects B cells, confounding factors in mouse model for inflammatory disease. This is widespread in laboratory mouse research facilities worldwide. This is how the MNV infected liver, you will find the coagulative necrosis. This also can be diagnosed by PCR method. At our place, we diagnose by both ELISA and PCR. Mouse hepatitis virus, morbidity and mortality is seen in infant as well as immunocompromised mice. Normal mice generally will not show that much changes, and they cause the wasting syndrome. The animal literally become cachexic, modulate immune response, changes in liver metabolism because it has the affinity to liver. It contaminates the biological products. It reduces incidence of diabetes in NOD mice, modulates T cell function, and this is highly contagious. When we have the, there is no barrier facilities, you consider your facility as infected unless you check by ELISA as well as PCR. Sometimes there are zero, what you call, similarity. And if you check by serology, ELISA, you may not be getting the positive result, but for that you have to go for the PCR method. The changes, growth changes are typical depressions because of the necrosis. In severe cases, the animal develop jaundice-like symptoms as well as the pathology. This is how the giant cells are seen, and the fibrous tissue development, proliferation. These are the photographs, images taken by me by mobile camera, and you can see the giant cells, necrosis, and multinucleate giant cells also. These are the typical pathognomic lesions, and this without the ELISA or PCR, we can diagnose them if we have the histopathology department. We diagnose by ELISA as well as PCR. This mouse was received for testing from outside, and we tested it for MHV, and it was positive. This is the positive control, and the sample from our facility, and then this testing sample, it was found positive. In the animal facility, when we are working, we must be observant. Sometime back, I mean a decade back, I got this florid papillomatous nude mouse, which I did the pathology, slide staining, and we can see the hyperkeratosis and finger-like projections. These are the papillomas. And with the Jackson laboratory, I collaborated, and I could find the, you can see the nice crystalline arrays. These are the viral particles, and this was published in Veterinary Pathology, official journal of American College of Veterinary Pathologists. And this is the first report of papillomavirus in the laboratory animals in the world. Mycoplasma is one of the again culprits. It influences carcinogenesis. Respiratory tract is the most affected. It damages of the airways and alveolar epithelial cells, mucus secretion, bronchitis, bronchiectasis, emphysema, abscess in the lung, and marked leukocytosis. So because of the secretions in the bronchioles, when you hold the mouse or rat near the ear, you can hear the gruntling sound, and that is one of the indications that the animals are suffering from mycoplasma. Immunology, diversified effects in the immune system, and another system is the reproductive system. It affects the genital tract. It has negative influence on in vitro and in vivo fertilization. So for Toru and group, it is important. On fetal development also, fetal losses and resorptions are seen, male and female infertility, and intrauterine infections of the embryos. When we encounter breeding depressions in the laboratory animals, we must consider mycoplasma as a culprit, because these mycoplasmas have predilection of site for the fallopian tubes. It goes there, sits and blocks the fallopian tubes, and that's how there is no fertilization and the reproduction is affected. This is how the hepatization and then abscesses in the lungs are seen. In case of mice also, you can see the linear changes. So the changes in case of mycoplasma is typical and symmetrical. It occurs when a pathologic process highlights an anatomic or physiologic subunits of the organ or tissue, often defined by vascular beds, lobules, ducts, airways, and tubules. So this is how the linear and symmetrical changes are seen. And here also you can see in this lane, and here also you can see. So this is a typical lesions of mycoplasma, and this is how histology reveals. So everything is consolidated. There is no air spaces to get the oxygenation, and that's how the animal struggle for oxygen, and there is labeled breathing. So we check it by, when we receive the samples, we check it by ELISA as well as PCR, and this is how you can see the 245 base pair product. And when we run all our samples in one go, and this is positive control and no sample positive. Helicobacter is also important. It colonizes in liver, colon, cecum, gallbladder, and stomach, and that's how cecum is the choice of organ collection for DNA RNA extraction. It is also liver tumors are because of the Helicobacter. Inflammatory large bowel disease also immunocompromised mice, kid mice. You can see these white discrete necrotis foci in case of Helicobacter, and this is because of the mild portal fibrosis. This can also be stained with Warthin stain, and it can be easily find out the Helicobacter. And if you see the lesions, sometimes you see the protruding anal mucosa. If you do the histology, it is the inflammatory bowels, and this can be easily identified. We diagnose by Helicobacter bilis as well as hepaticus by PCR method, and we also have bacterial infections in the animal facilities. So Pseudomonas, which can be diagnosed by conventional media plates, and these are the biochemical tests which are in conjunction with the conventional can be conducted, but the confirmation can always be by PCR method. So we also do the PCR to check the Pseudomonas. In hamsters, there are few listing by the Phalasa, and Clostridium difficile is one of the culprit because it has the hemorrhagic, digestive system, and you can see the villi and the hemorrhages. In hamster, again LCMV, similar zoonotic importance. You can see the meningitis, lymphocytic meningitis, as well as the chronic glomerulonephritis. This is very rare, but which is infectious hamster polyomavirus, and you can see the glandular, polyps, and this is because of the trichoethylene epitheliomas. Helicobacter again in case of hamsters, and you can see the bile duct proliferations. Teaser disease again similar to the mice and rats. Salmonella is one of the again zoonotic disease in case of hamsters, and it produces thrombosis of pulmonary as well as liver venules, multifocal necrosis of the liver as seen in the photographs, and diagnosis can be by culture as well as PCR. However, for Salmonella, it takes long time to diagnose, to incubate, as well as the treatment is also very long, similar way to the human patients. In case of rabbits, also several viral, bacterial, as well as fungal diseases are there. Some of the important are Pachyurella. You see the conjunctival hyperemia with development of chronic epiphora and inflammation. Gram-negative short bacillus, coccobacillus, and this can be by culture, ELISA as well as PCR. We can diagnose it, and this is how the lungs can be seen in case of rabbit Pachyurella. This is very normal. Hepatic coccidia is very normal. Eimeria steady is the lesion. You can see these are lesion because of the cholangiol cell proliferation. This is how we can see them. And in chronic cases, you can see the liver is like this because of the very high concentration of the Eimeria inside the hepatocytes. Encephalotoonosis, this is zoonotic to humans, and the cunicula you can see in the eye. This affects the eye, and it reduces the breeding as well as performance, and also affects the welfare. In case of zebrafish, also there are quite a few infectious diseases. Like if you see the microsporidiasis, you can see this spinal curvature. Whenever you see such curvature, of course there are some bone deformity also, but when you see this one, you must check for pseudoloma neurophilia. We do receive sometimes such samples, and I have seen the transmission is vertical as well as horizontal transmission, and When we receive samples, we tried to diagnose it for the pseudoloma, and we could succeed by developing the PCR band. So these are some of the samples we tested. Similar way, there is tuberculosis in case of zebrafish, and they develop granulomas. These are caused by Mycobacterium marinum, which shows great similarity with human granulomas formed after infection with the Mycoplasma tuberculosis in humans. You can see the tubercular structure, and this is how the lesions are seen throughout the body. This can be, uh, stained and can be easily identified also. But of course, you need the PCR method. There are some of the parasites which are important in case of laboratory animals. Some of them are very difficult to get rid of, but we must diagnose them. This is how the Syphacia pinworm can be diagnosed. This is the unique method. You can see the cellophane tape method. With stool sample, it is very difficult. You may not get the eggs because the females has tendency to lay the eggs near the anal region towards the rectum, and they are sticking to this perianal region, and only cellophane tape can be used for diagnosing. And you can take this cellophane tape and you touch here gently and push on the slide. You can identify the banana shaped pinworm eggs. This is from our one of the publication, and it is very easy to diagnose when you have the parasite. When you see the esophageal valve round shape, you can see that this is the Syphacia. In case of aspicularis, it is elongated, and this is the high power view of the esophagus in case of Syphacia. And another feature is the tail of the Syphacia females animals is very elongated, pointed, as compared to the spicularis, which is blunt. So these are two very important features to diagnose. Although there are other methods, PCR methods also available to diagnose the endoparasites. And you will be surprised to see that one female Syphacia can lay hundreds of eggs. This is how we captured and published some time back, a decade back. And one egg is responsible to give rise to the infection in the colony, and this is very difficult to get rid of. The treatment is very harsh and continue for at least a month period, then only you can get rid of the pinworm. We also published the Klebsiella in case of this is a kidney parasites, and this was published in way back in '99 in our facility, and I could also get rid of this one by sulfa drugs. Klebsiella, which is a kidney coccidia, not considered as pathogenic, but it affects weanling mice, and a lot of load is there, susceptible to other pathogens because of the kidney parasite. Males are more heavily parasitic than females. It influences vaccine efficacy. Villus to Krebs ratio are reduced, and variable number of lymphocytes may be seen. Sometimes it is zoonotic. Clinical signs are non-specific and includes weight loss, stunted growth, rough coat, and enlarged abdomen. Immunocompromised develop diarrhea and death, and decreased body weight and reproduction, as well as lifespan because of the Klebsiella, which is kidney coccidia and very rare. Now, knowing that all these variables are there, infectious variables, how to balance the animal research and the quality of the animal research. So, as I said earlier, we have to identify and deal the variable. There are variables, other environmental as well as genetic, but how to deal with this is by microbiological control. Unless we have the microbiology laboratory, we cannot really find out our quality of animal. So there are methods like conventional. We can have the stool samples like tape test or media, conventional media, as well as special media. For example, Salmonella shigella agar, which is the only option for this one, or for Pachyurella bovis, we can have the blood agar, wherein it takes long time to culture. We also take advantage of biochemical tests to zero down on the differential diagnosis in different bacterial organisms. And when we need to do antibiotic treatment, although it is not advisable, but some of the experimental animals, we cannot afford to lose them. So during the long-term experimentation, we may have to do the treatment. And for that, unless we do the uh, antibiotic sensitivity, we cannot institute the antibiotic treatment. We can take advantage of the ready-made ELISA kits available commercially, and of course, PCR is the best method to diagnose these infections. So this is how we can culture in normal media, and as I shown the biochemical markers, biochemical test. And when we are doing the antibacterial treatment, we have to do it on for bacteria, of course, on the media, and for that we have to do the antibiotic sensitivity test. And based on the zone of inhibition, we have to select the antibiotic, and then only we must do it. Otherwise, there are a lot of rampant use of antibiotics, and the animal may develop the resistance. They may not respond to the treatment. So unless you do the antibiotic sensitivity, it is not advisable to go for the antibiotic treatment. And of course, as I show, the ELISA is one of the tool to diagnose the animals commercially if we don't have the in-house laboratory to diagnose. And of course, PCR is a gold standard and a confirmatory test to diagnose the infections in the laboratory animal facilities. And to summarize the talk, use of laboratory animals is inevitable in laboratory research. Laboratory animals are prone to variety of infections like we do, and it causes interference in some of the research as I shown here. But quality animal production is possible through microbiological monitoring, and that's how we can achieve reproducible results, and also 3 R's of the Russell and breath as well as the animal welfare. With this, this is my institute entrance and campus, and thank you for your patience.
Thank you very much, Dr. Arvind Ingle, for that engaging presentation. We would now like to invite Dr. Hasanti Ratnadivakara to present the token of appreciation to Dr. Arvind Ingle. Once again, thank you very much for Dr. Arvind Ingle for that engaging presentation. And thank you, Dr. Hasanti. Ladies and gentlemen, we do have one more question for honest and attentive students to answer, not the high-end technology or chatbots. So the question will be, what is meant by microphysiological system? I'm going to repeat the question. What is meant by microphysiological systems? Okay. Yes. Yes, we take that as the correct answer. Congratulations, and we invite the president of SLALAS, Dr. Sachini Amarasekara, to award the gift for our winner. Yes. It's being his third gift for the session. Thank you very much, Dr. Amarasekara, and congratulations for the third time. Now it's our pleasure to hear from the winner from the Sri Lanka Association for Laboratory Animal Science Oratorical Competition. This talented individual has demonstrated exceptional skills in articulating the importance of laboratory animal science and the power of communication in driving the progress in our field. Let us give a warm round of applause as Miss K.D. Manchi Mantila will take the stage from now onwards. Over to you, Miss Manti.
First of all, I would like to thank SLALAS for giving me this wonderful opportunity to stand in front of such honest gathering. Today, I am going to tell a few words of role of laboratory animals in precision medicine. First of all, I would like to give a brief introduction of what is precision medicine. Precision medicine is a form of medicine that use information about person, gene, protein, environment, and life cycle to prevent, diagnose, or treat a disease. Laboratory animals like rat, mice, pigs, zebrafish does a crucial role on it. Why we need precision medicine? Even every person is unique, and also they are different in environment, life cycle, and also the genetics. That's why we need a specific, we are specific to person. Precision medicine is specially used for identify the cancers or treat the cancers. For example, some cancer drugs works only for the patient with specific genetic mutations such as HER2 positive breast cancers. Today, breast cancer is the most commonest cancer in women, so we have to treat those cancers for specifically to reduce the death rate. On this, I read some articles about the cancer. According to the breast cancers, they are used many articles use mice to identify these cancers, and also mice used for the identify the liver tumors too. We can use different animal models like NSG mice for the oncological research studies. An animal model can play an essential role of the precision medicine, but there is a task to identify the relevant or the suitable animal model to our research, because when they are going to do a tuberculosis in transplantation and precision medicine, they identify each animal model have own advantages and disadvantages too. Therefore, the relevant animal model taken is somewhat difficult task. Therefore, there are animal models, we have to reduce their pain, and also we have to reduce the number of animals that we taken for the researches. There are many researchers using animals for the precision medicine. Use of animal model, we can able to get a more further advanced treatments in medicine. Thank you. And again, I especially thank for the SLALAS giving me this wonderful opportunity and unforgettable memories. Thank you.
Thank you very much, Ms. Manji. And to present the award for the winner of the rhetorical competition of Sri Lanka Association for Laboratory Animal Science, I would like to invite Dr. Suresh Pusala, the founder of Akasta Health and Oncoseek Bio Private Limited. Once again, ladies and gentlemen, give a huge round of applause to the winner of the SLALAS rhetorical competition, Ms. K.D. Manji Mantila. Thank you very much, Dr. Suresh, and congratulations to our winner. Now we are excited to invite you to explore the poster session, where you'll have the chance to engage with the cutting-edge research in laboratory animal science. This is a wonderful opportunity to interact with fellow researchers, ask questions, and discover the innovative works which been done in our field. Don't miss out this chance and be inspired and connect with the new ideas. Additionally, we would like to inform all the poster presenters that the evaluation panel will be visiting the poster areas to assist the presentations. So please ensure that you are present at your poster and ready to discuss your work when the panel arrives. Now I would like to invite Dr. Suresh Pusala, the founder of Akasta Health and Oncoseek Bio Private Limited, to chair the poster presentation session. Over to you, sir.
Here at the back of the room. They will be available for all of you to visit. And there are 3 judges who have been identified who will be visiting and talking to the presenter along with the speaker in front of this poster. So please try to answer as many questions as possible. And the print of the poster to the bottom is a little bit small and also close to the floor. But the good news is these posters can be lifted. I tried it yesterday and very easily with one hand. It's less than 100 grams. So I think you should probably try to pick it up from the backside and also explain it with your finger, pointing it to the judge especially. For the rest of them, I think they can make some effort, but I think with the judges, you need to be a little bit more careful about how you present yourself on the results. So this will be open for 30 minutes. From now, and post that there is lunch. So please enjoy the posters and join us for lunch in the cafeteria downstairs. Thank you.
Welcome back, everyone. We hope you had a lovely lunch and are feeling rejuvenated. We are about to dive back into our engaging scientific sessions with even more brilliant speakers lined up. Let's continue the journey of discovery and collaboration. We are pleased to welcome Professor Pascal J. Lafontaine from the Biology Department, Noise Science Center, Grinnell College, USA. Professor Lafontaine will speak on fish models of myocardial infarction and heart regeneration. I would like to call upon Dr. Prasaditha Silva to introduce Professor Lafontaine. Over to you, madam.
Thank you. So it's my honor to introduce Professor Lafontaine to you. Professor Pascal Lafontaine is professor in biology at Grinnell College, emeritus professor of biology at DePaul University, and affiliated professor in the genetics, development, and cell biology department at Iowa State University. He has broad training in cell biology, physiology, tissue remodeling, heart development, and regeneration. He earned a BS in biological engineering from Cornell University and MS in biomedical engineering
from the Harvard Graduate Center, Rensselaer. I'm sorry if I pronounced wrong. Yes, and Polytechnic Institute. He completed his PhD in biomedical science in the DeBakey Heart Center at the Baylor College of Medicine, where he studied the role of inflammatory cells and fibroblasts in the repair and remodeling, especially on myocardial infarction. He was a postdoctoral fellow at the Wells Pediatrics Research Center, Indiana University School of Medicine, where he studied cell cycle reactivation in differentiated cardiac myocytes and the contribution of stem cells to cardiac regeneration. He has studied heart development, repair, and regeneration in fish models for the past 2 decades. Professor Lafferton has been a visiting scholar at the Cardiovascular Research Center of the Massachusetts General Hospital and Iowa State University. He is a Fulbright USA Sri Lanka scholar based in the Zoology Department at University of Peradeniya for the 2024-2025 academic year, where he is studying the development and ecophysiology of cement glands in Devario species. So, dear Professor Pascal, over to you. Thank you.
Thank you. So thank you very much for the introduction. I do want to thank SLALAS for the invitation. I also like to thank, I'm based in Peradeniya, but I have to say that I feel that I've been adopted in Colombo. So I'm kind of the adopted son. So I want to thank some people for that. I would thank Dr. Prasadi, Dr. Mangala, also Dr. Kalpani that invited me for the last meeting, and Dr. Sakine. So as was said, I'm the Fulbright scholar, or a Fulbright scholar, one of the 3 Fulbright scholars currently in Sri Lanka, and I'm going to be here until July. This is my 4th month already. And I'm almost a local because at least once a week somebody comes to me and starts to speak to me in Sinhala. And then when I say, well, I wish I could speak Sinhala, but I'm not there yet. I'm taking lessons, but I can't have a conversation yet. But in any way, my work here is intended to study cement gland in Devario. I would love at some point to actually give a talk on that. But on the side, what I typically do is study heart development and heart regeneration, and in particular in a species that is native of Sri Lanka. So that's what I'm going to talk to you about. Most of the work that I'm going to talk about that you're going to see were done at DePaul University. I'm no longer there, but I'm an emeritus there. And that's has been the focus of my work for the past 2 decades, understanding how the heart responds to damage, what are the limitations that prevent humans to regenerate their hearts, and also try to learn from species that can actually perform this, near miracle in creating new cardiac tissue that beats. So at some point since I'm Sri Lanka, I wanted to look at the epidemiology. So, from the US or Europe perspective, we used to think of cardiac diseases or cardiovascular diseases as something that tend to happen in mostly developed countries, and that's also tied to changes in diet. So in the US, you can see some numbers, about 600,000 people would have a heart attack every year. And then about a third of that number have a second heart attack or sometimes a third heart attack a year. And quite a lot of those heart attacks are silent. You might actually have one and you actually never noticed. You might notice that over time you're losing your capacity to do exercise and things like that. But there's no significant symptom that would actually send you to the hospital. So some other, kind of scary data that in average every 34 seconds, somebody dies in the US of cardiovascular disease. We also know that most people that are at high risk of cardiovascular disease or that have had a heart attack have, their cholesterol level pretty high, especially their LDL, which is the low density lipoprotein. That is what we call the bad cholesterol. But beyond the US, Europe, and Japan, and other places that are more developed, you can also see the numbers are pretty high globally. So cardiovascular disease is no longer a developing country or a developed country disease. It's broad, it's just global. I was surprised to learn that actually cardiovascular disease is pretty significant in Sri Lanka. I was telling Dr. Prasadi that I feel bad that I'm going to talk about cardiac disease after lunch. So people might be thinking, what did I eat today? So you're going to see here, which is surprising, that 40% of all death in Sri Lanka can be attributed to cardiovascular diseases. So that's very significant. There's a couple of studies actually done by, I guess, scholars that are in Sri Lanka here that look at some of the data, and cardiovascular disease is on the rise, continue to rise in Sri Lanka. I did notice going down Duplication Road that there's a lot of Burger Kings, Subways, KFCs. I was at the Sematakumbura bus station a couple of days ago, and then there's a Pizza Hut, and I think it says favorite home food, something like that. Is that what it says? Which was surprising and ironic, because it's not home food, right? So people that have high LDL are at risk of, I was trying to find it. Here we go. Yeah. To build plaque in their vessels, and then if one of the plaque is in one of the descending arteries in the heart, the plaque becomes unstable. It dislodges, go down, and block the major vessel. You deprive the rest of the heart downstream of oxygen, and when the area becomes anoxic, so no oxygen at all, you develop an infarct, so cell die, you get necrosis. And here in a cross section, this is a typical area that is infarcted, that is dead. And then next to it, there are tissue that are what called at risk that also can die over time. And you have no ability as human to regrow muscle cells, so what you develop basically is a big scar that does not have the ability to perform the contractile function that would allow you to get blood out of the heart. But if you look at other species, there's quite a few of them that have this ability to regenerate the heart if the heart is damaged. So you have some newts. You have the axolotls. This is a Mexican axolotl that you find south of Mexico City. They are an endangered species right now, but there are some labs that grows them, and you can get them from labs. And the zebrafish in 2003, there was a first study that shows that the zebrafish can regenerate its heart after damage. And about 10 years ago, maybe 14 years ago now, there's a group that was in Atlanta that shows that a neonatal mouse, up to 7 days, can regenerate part of their heart if you damage it, but after 7 days, it can no longer do it. So a lot of studies have followed that, where we've been comparing species where the heart can or cannot regenerate. So here, zebrafish can regenerate. Medaka cannot develop a scar. Adult mouse, of course, cannot. I don't remember. I think the killifish cannot either. The axolotl can. The newt can. And there's another Mexican contribution. So this is Astyanax mexicanus, which is the Mexican blind cavefish. So the cave morph is blind. You see there's no eye. The eye is underdeveloped or has disappeared because they live in dark caves, so they don't actually need eyes. But the surface one actually can regenerate. So there's a lot of interesting area that can be studied. Fundamentally, what limits human, mammal, adult ability to regenerate has to do with the fact that the cardiac myocyte, which is the muscle that beats, as you're in utero, you can divide. That's when you can enter the cell cycle and exit the cell cycle and re-enter the cell cycle and exit the cell cycle. But once you're born, very soon after, those cardiac myocytes no longer can divide. So the number of cells that you have in your heart at some point in your early years, those are the same number that you're going to have the rest of your life. So you're wondering how come your heart grow bigger? The only way your heart goes bigger is each cell actually grows in size. So they grow by hypertrophy. So if you lose any cell here, you can't replace them. But we can clearly hypothesize, and we have evidence that in fish and amphibians, they have the capacity not only to do hypertrophy, but they can do hyperplasia. So that's when you can re-enter the cell cycle. I'm open to answer any question if you have question while I'm talking. Okay, so this is the basic universal response to damage. So small damage here, a little bit more significant damage there. This is the response to damage. This is a big scar here that you have. It's big enough that you call it a keloid. This probably will heal fine. Here you can see the thumb over time. It takes time, might reconstitute itself. And typically from injury, what you tend to have is, of course, hemostasis. You stop blood flow. You have a period of inflammation, cell proliferation. And this area here that we call remodeling is you have cells like endothelial cells that form blood vessel, fibroblasts that makes collagen. And the outcome of the injury will depend on which part of this wins the battle. Too much collagen, scar, more blood vessel, more cell cycle, no scar, tissue regenerate. So similarly for the heart, that's what's going to happen. But in the heart, what tends to happen is fibroblasts proliferate, myofibroblasts, a lot of collagen, angiogenesis, new blood vessels fail, but also the fact that those cells, the beating cell, cannot divide leads to what we call heart failure, when basically you get to a point where your heart can only pump 20% of what it receives. So with what we call ejection fraction less than 20, you're like in heart failure. The approach to remedy this problem are twofold. At least you can think of two possibilities. You can either identify stem cells or various sources that you might either grow in the lab, differentiate to cardiac myocyte, and put back in the tissue, or you can put the stem cell directly into the heart and hypothesize that signal around will actually direct them to become cardiac myocyte. The other approach is the cell that cannot divide, you find a way to force them into cell division so they can make new cells. Okay. So my lab have used zebrafish, and this is the giant Daniel from Sri Lanka, and then and goldfish as a way to compare the ability to regenerate and the method by which they regenerate. So those are some. All the students are undergrad students. They are from first year, first semester to fourth year. Okay. So we use fish. How do you do a heart attack analog in an animal? So if you're a mouse, you can open the chest, identify a coronary, left descending coronary artery, take a string, block the vessel. You can block it permanently, or you can tie it for about an hour and then reopen it. So that will be equivalent of somebody having a heart attack, goes to the hospital, they get, the clot being removed and they get reperfusion. Now, if you have a fish, the first study in fish was done by Ken Parsons now at Duke, where he resected a piece of the apex of the heart. And then after that, some years later, people have started to use cryoinjury. They use a probe, they cool it down in liquid nitrogen, touch the apex, and get an area that is diseased. And then after that, new technique can use genetic ablation, where you can express things like nitroreductase directly into cardiac myocyte that you can trigger to kill cell. This is more broad. At the bottom here is what we do in the lab, which is a cauterization. It's a probe, but we heat it up. And in this case, this is a goldfish upside down in the little sponge surgical table where we can do the surgery. This diagram shows kind of two processes. So this is the fish. You open the window, see the heart. This is about 1 millimeter across, so it's very small. You have to use a microscope, and you really have to have steady hands. No coffee before the surgery. And then you can resect the apex, or you can use a probe to either cool or heat this area to cause the damage. Any question? So this is a diagram of a probe being made, Benson. So it's very simple. Everybody can do that. You don't need any sophisticated equipment. This is a zebrafish here. This is one of my students' finger, maybe mine. And then that's how we set it up. And this is a zebrafish study that was published now 4 years now, where we can demonstrate that you see this area here, or the damaged area. These tend to average about 20% of the ventricle, and this at 24 hours. And then what you can see over time, this area, slowly the dark area goes away. So this is all dead tissue, but you can see some whitish more tissue coming. This is new muscle cell coming up, and if you look closely, there are little blood vessels here. And then if you look at about 30 days, it's mostly regenerated, and by 120 days, those hearts look like heart that never been damaged, except you can recognize a damaged regenerated heart because usually there might be a little notch like this that you would not find in a non-injured heart. I'll show one more example for the zebrafish, where at that time we're interested to see if reinnervation comes back after damage. So this is the green line represent immunofluorescence of nerve using acetylated tubulin or ZN12. This is cauterized heart, where you can see the area that is damaged. There's no nerve at all, and this is all the innervation on top here. And then over time, the nerves slowly come back, and at 120 days, the heart is fully innervated. Okay, so that was zebrafish. Now, for Sri Lanka, this is the giant Daniel, and I think it has a different name here. I think it's called Salia? Salia. Okay. So this is Salia. It's bigger than the zip. What is it? Savalia. Savalia or Salia? So this is Savalia or Salia. So this actually species has been used before. There's about 40 plus. Those are Devario. This is Daniel. This used to be classified as Daniel, but then they got reclassified. They are definitely different genus. There's about 40 plus Devario species. You find them in India, Sri Lanka, Nepal. And they've been used to study adipose tissue, nose, muscle biology, vision, swimming, and biomechanics. In Sri Lanka, there are about 6 Devario species that have been documented. I don't know if there's more or less. It's a little bit controversial, but this is the Devario malabaricus. That's what we use. And then there's the Devario Patirana, and then there's the Memorialist, and then there's a couple of the others that I don't have a picture. So my interest then, before even thinking about or coming to Sri Lanka, is I was interested in that Devario because this is a SEM looking at the heart of the Devario. This is the heart of a zebrafish, an adult zebrafish. You can see the heart is very spongy, and then there's no thick wall. It's a very thin wall here. So my question was, does this really fully represent a human heart? Because a human heart is the wall is very thick, right? So then I wanted to use a fish. I wanted to know if this specific parameter could actually have an effect on the ability of the heart to regenerate. So I decided to go with the giant Daniel because you can see there's a lot more muscle. It's a more dense heart. Of course, they are bigger fish. They are more sporty, right? They live in fast river. They swim pretty hard, so they need the strength in the muscle to be able to pump the blood that they need. And then if you look closely at the wall is actually a bit thick. You have blood vessel in there. So this is the thick wall here, and this is the spongy heart. Okay, so we had a hypothesis, and whether this fish could be used as a model for cardiac remodeling and regeneration. So we developed this cautery injury. Now this is what we call plastic section stained with toluidine blue. You can see here, this is the bulbous, this is the aorta of the fish, this is the whole ventricle, but you can see here, this is the part that has been damaged. If you zoom in doing electron microscopy in the undamaged area, you can see the nice actin myosin pattern filament with zebin and sarcomere. I'm biased, but this is gorgeous. And then if you look at the area that is damaged, you see all the actin myosin structures are gone. You can see some cells that represent inflammatory cell. And then if you look at what we call the border zone here, you see an area where completely disorganized, and this area is not as well as organized as this, okay? So if you follow those fish over time, grossly what you can see, just like you see in the zebrafish, that at 3 days, this is about 20% of the heart is damaged, at least look damaged. And here at 7 days, this is 14 days, you can see the damage area decreases slowly. By 21 days, you visibly there's this, and then by 45 days, the heart look like a non-injured heart. Now, to confirm that, you do section. Here we stain with trichrome stain. The blue is for collagen. You can see in the ventricle there's not much collagen at all, but in the damaged heart, at 7 days, a lot of dark tissue here, which is damaged tissue. You can see a little bit of blue here. Now you can see the damaged tissue goes away, but you see more blue. So there's a lot of collagen being deposited. But if you fast forward 60 days, you see that the muscle is back, the wall is back, although you find some traces of collagen here. And if you look like at 180 days, then that collagen is gone. So this fish is able to generate collagen and almost a scar just like human, but also has the ability to resorb the collagen over time. And that could be supported by those cells actually dividing. So the other part of remodeling that we wanted to consider was that first phase, which is inflammation. So do we have inflammation after the injury? Yes. This is myeloperoxidase staining, where the black area represent enzyme that is present in neutrophil and white blood cell. So 24 hours, you see a lot of dark, so a lot of neutrophil here. And then 14 days is almost gone, and then you don't see much inflammation late, but strong inflammation in the beginning. Those are just higher magnification or zoom on the area that are damaged. Here you can see there are little dots of white blood cells scattered all over, and then here the white blood cells are gone. So at some point the inflammation goes away. Those transfusion electron micrographs show different type of white blood cell you can find, and you can see inflammation comes up, stay, and then goes away. If we look at more closely at collagen deposition, zooming, you can see a lot of blue here, a lot more blue here at 14 days, but you can see at 60 days, there are some blue, but you can see the muscle that wasn't there is back here. Okay, so it regenerates collagen get deposited, a little bit slower goes away. Those TEM also show the different type of collagen. This is mostly collagen type 1 that you find in the area. If we do, somebody did some lectin staining. We also use lectin here to reveal the blood vessels in the giant Daniel heart. You can see it's very highly vascularized. And then after the damage here, there's no blood vessel at the edge here, but over time, the blood vessel also come back. And finally, do the cardiac myocyte divide. So this is 3 different assays that shows here. This is pCNA, histochemistry. In the black dot represent cells that are proliferating. Here is EDU incorporation in red. Green represent myosin for muscle, and you can see the red nuclei, which are cell in the cell cycle within this myocyte population. And to confirm that further, we did some very old technique called autoradiography, where we use radioactive thymidine, very thin section, about 1 micron section, and then we develop over time, and you can see those little grain over the myocyte that indicate that those myocytes are in the cell cycle. And finally, if we look at a nice control here at 14 days after cautery, this is where you can see ZBAN, a little bit of actinoisin filament. This is a heart that is slowly regenerating and rebuilding the cardiac myocyte until you have fully functional cardiac myocytes. So our conclusion is that the Sri Lankan giant Daniel, basically there's a permissive form of remodeling that does not lead to scar, but that include inflammation, resolution, scar removal, angiogenesis, new blood vessel, and cardiomyocyte cell proliferation to support heart generation in the giant Daniel. Okay, I'd like to thank the students that has done those work. So those are the students that did most of the giant Daniel work. Those are the students in the middle here, Tina, Julia, Maria, and Adedoin, that did the goldfish work, if you're interested in reading the whole thing. And then those are some of the students that have done the zebrafish work that I showed you earlier. Okay, if you happen to have your QR code reader and you can look at this, you'll get all the papers are public, available now, so you can always go to them and read them if you'd like to. Okay, so as I'm ending, we've also studied the development of the giant Daniel. So this is 1 hour, 8 cell stage. This is 8 hour, like 90% epibole, and then here you get segmentation and so on. And this is what we came across, some interesting behavior. This is how we breed them. So you see larvae, a couple of days old, unusually standing up. This is the surface of the water. Here, larvae still kind of standing up against the wall of the tank. This is more puzzling, where the larvae are in line, not attached to anything. This is the surface of the water. And those are some of the behavior that puzzled us for a long time. Once they hatch, they start swimming upstream, and they stop swimming once they attach to a substrate. You can see we wanted to replicate what happened in Sri Lanka, so we brought some moss into the lab, put in the tank, and they attach. I want you to pay attention to this guy. Oh, it happened already? Oh, it didn't move. Right. So the movie didn't work. But if it worked, this fish would detach and then swim back and attach itself again. Okay. And the last one, hopefully this one will work. People find this really fun. So we're trying to mimic Sri Lanka in the lab. So we put some rock and then move the water, and you can see them swinging. Okay, so all this leads you to believe something is attaching them. What attaching them is not visible, but we figured out that it's because the refractive index of the whatever compound was the same as water. That's why you can't see it. So we use lectin again because we hypothesized that if there's a glue, for example, it will be highly glycosylated. So lectin recognizes carbohydrate on surfaces of cells. So we used that, and we were able to identify very highly expressed or reactive to WGA lectin right on the top of the head. You can see this little transparent structure here. So in a way, we've discovered that these fish have what we call a cement gland. If we do SCM at 72 hours, this is the two eyes here, this is the mouth, the nose, and here you have that nice little cup that looks like a sunflower or cauliflower or broccoli that has been flattened. Nice petal-like epithelial cells surrounding it. And within the core of the gland, there are cells that you can't fully see here, but you can see those little bubbles, and those bubbles are glue that is being expressed by the cell that actually makes the glue. To confirm that further, take a tweezer, touch, and then move. You can see the string of glue coming out. And then you can see we can stretch it, and it can stretch pretty far. Okay. So my question is that I would love to talk to people interested in this and collaborate maybe is, do all the Sri Lankan Devario have this gland? What do they use it for? By the way, this gland disappears after 2 weeks. The fish only use it in the lab for 5 days. They stand up this way until day 5, and day 6 they detach, and then they start swimming. Okay. After working 20 plus years on heart regeneration work, we got paper published, but we never made a cover of a magazine. And then we found this gland, and we were lucky enough to be featured in the cover of Developmental Dynamics. So this is the gland here with nerves innervating it. This is another view of the gland where the nerves and the trigeminal nerve are innervating it. There's a lot more to say about this. I would be happy to discuss that, different forum, different time, to whoever want to talk about it. This is the last picture showing the secretory cell with the bubble of glue being produced and all the granule that contain that glue. So primary collaborators, Alan Burns, Yusuf Houston, and Sam Winitoma Michael at the POR, and then a lot of funding from NSF, had a funding from NIH, fellowship from Reich, Buller Foundation Fund that allowed me to start a biomedical imaging center, and then of course Fulbright for the work that I'm hoping to do in Sri Lanka. Okay, so I got the wrong. So those are the two students that mostly work on the cement gland work. All right, thank you.
Thank you very much, Professor Pascal J. Laffonenton, for your fascinating presentation. We would now like to invite Dr. Prasadi De Silva to present the token of appreciation to Professor Laffonenton. Thank you very much once again, Professor Pascal and Dr. Prasadi. Ladies and gentlemen, as a part of today's event, we have sent an open invitation for the roundtable discussion on laboratory animal science and the significance of media role in it. We all know communication plays a crucial role in laboratory animal sciences, ensuring that both scientific community and the public have a clear understanding of the importance and the ethical considerations of animal-based research. This session aims to foster an open dialogue between laboratory animal scientists and media professionals to promote transparency, ethical practices, and responsible coverage of media research. It also seeks to address public concerns and ensure accurate media reporting on the critical role of animal research that plays in advancing medicine and science. At this moment, we would like to take this opportunity to confirm if there are any representatives from local media institutions present with here today. If any members of the media are here, we would be delighted to begin the media segment now. We are going to open the floor, so you can ask any question in Sinhala as well. නිවැරදි සහ නිර්වද්‍ය මාධ්‍ය භාවිතය විශේෂයෙන්ම සත්ව විද්‍යා පරීක්ෂණ ක්ෂේත්‍රයේදී අත්‍යවශ්‍ය වෙනවා. ඒ සඳහා සුවිශේෂී අවස්ථාවක් අපි අද ඔබට හදලා දීලා තියෙනවා. අපිට දැනගැනීමට අවශ්‍ය වෙන්නේ යම්කිසි ශ්‍රී ලාංකීය මාධ්‍ය ආයතනයක් නියෝජනය කරමින් යම්කිසි නියෝජිතවරයෙක් මෙම ස්ථානය සඳහා පැමිණ සිටිනවාද නැද්ද යන වග. Well, if there are no media representatives present, we would like to take advantage of this time by opening the floor to questions from the students to the audience. This is a rare and invaluable opportunity for you to engage directly with our speakers, an opportunity you may never get again. So please feel free to ask any question you have about this work and expertise or the topics that discussed today. This Q&A session will continue until 2:30 p.m., after which we will begin the scientific session as scheduled. All the participants, the floor is open for you all to meet all our distinguished speakers, once in a lifetime opportunity, and come here and ask the questions that you all have regarding their research work. Well, you don't need to come to the mic to network. You can go to your professors and ask questions.
So this question is to Professor Pascal. So you mentioned something about the semen gland for the Deverio. So what is the how can I ask that? What is the special characteristic of the semen gland, or is that just an anatomical structure of that Deverio?
So, for example, how it actually developed is pretty interesting. Um, the Devario is not the only species that have this gland. So there are other species that have uh, analogous gland in some ways. Like if you think about, let's say, barnacles, that attach to boat and piers, they have some kind of organ that secrete a cement that will allow them to attach to things for the rest of their lives, right? There is a cement gland that has been found in some amphibians like Xenopus. So Xenopus have a cement gland, but their cement gland tend to be kind of on the chin. Okay. I don't think people have studied how long they stay when they come. I mean, there's some study that talks about it in the beginning, but not really in detail. Okay. There are some other fish in Brazil and Africa where cement glands have been identified. In fact, there is angelfish have 3 pairs of cement glands that are bilateral on each side of the head, and in fact, one of my current colleagues at Peradenia is Shalika Kumberagama, that studies the cement gland on the zebrafish. And then one of her students, Rishani, which I worked with this past couple of weeks, is going to defend her work on the cement gland 2 days from now. The giant danio, at least this species, we found out that they have a cement gland. We don't know if the other Devarios have it, or maybe there are other fish also within Sri Lanka that has it. There's a number of unanswered questions. What is the nature of that glue? What makes it so flexible? What's the biochemical components that make it? Functionally, it's clear, at least from the lab, that they seem to need the gland attached until they're able to swim. And 2 things are required for them to swim. One, the swim bladder has to be fully inflated. And second, the pectoral fins have to develop enough that they can move and also to propulse themselves. So it seems to us in preliminary data that those 2 things happen. And when those 2 things happen, they actually detach. Okay. I give long answer. But I think it's fascinating. That's why I give long answers. There's a particular thing for developmental biologists, which is biased, really cool. The structure that you see that looks like one structure actually start as 2 separate structures. And they start like other fish bilaterally. And it seems to us in data that we have, during the process of what's called neurulation, when the ectoderm kind of caves in and folds to form the brain and the spinal cord, during that period, the 2 fields of the secondary cell migrate together and fuse to make one. So this is unique on all the cement glands that have been discovered so far. Okay, and to test that, we actually can inhibit a particular molecule called non-muscle myosin, and if we block it, we actually can create larvae with 2 glands as opposed to 1 gland. Does that answer your question?
Yeah.
Do you have other question?
No.
I'm more nervous than you are, so don't worry.
No, I want to know that, does that have any significance? I mean, can that be used in any other factors like the cement gland? So apart from the fish, like for any other uses? I mean.
So there's other avenue of research. So for example, one of my interests is understand the biochemical component, because if you think about in medicine, surgery, right? Usually after surgery you have to seal, you can use suture, but sometimes now depending on the injury you can use like collagen, as a way to seal. So to me, if I think of it more in terms of biophysics and medical application, this is a glue that works underwater, right? the glue business overall is like 40 billion a year. So imagine if you can figure out how to make this glue that works very well underwater. Yeah. Maybe you can open a startup company maybe a couple of years from now to explore that. In terms of, I guess, ecological physiology, it seems like they might need, because they live in those fast stream, in the plateaus, in the mountain of Sri Lanka, they might need this to remain in their natural environment so they are not carried away. Yes.
May I make a comment on this, Professor Pascal? I think that the role of cement gland is kind of survival of the larvae, because first 3 to 4 days they don't need food, so they stay in one place, and maybe it's a natural phenomena that to resist the current, so they can stick to one place and survive. And I'm not sure about the cell structure, whether they have goblet cells or some mucus secreting cells inside. If so, it has some function on innate immunity as well. That's just my thought, but it's really interesting to
This is a
study more.
fantastic thought. This also revealed to you that there's a lot of things that I don't know. I'm not an evolutionary biologist. I'm not even a developmental biologist. This is something that I just bumped into and I studied to learn about it. So what I'm telling you is things that I've learned that I didn't study ever. It's just like I'm learning as I go. Okay.
First of all, thank you for giving this valuable opportunity. My question is for Professor Pascal. Sir, can I know, are there any other alternative methods that we can use for the cardiac tissue regeneration in, especially in the indigenous medical types in your country? Are there any tests have done, or are there any methods that you have used in your country for the cardiac tissue regeneration?
So just to be clear, are you talking about different animal models or approaches to regeneration, or both? I'm trying to understand.
I think, Professor, the question is like, now there are natural alternatives that in Ayurvedic and traditional medicine that we use to regenerate, that they say that they can regenerate these tissues. So have
I'd
they
love
been
to see
tested?
that.
Yeah. Have it been tested in vitro or in lab setting using zebrafish or?
Let's say in mouse, one of the approaches, I worked in a lab that actually tried to reactivate the cell cycle in cardiac myocyte by expressing, I don't know if you know about those proteins, cyclin and cyclin-dependent kinases. So if you're able to transgenetically express those proteins, you actually can force those cells into the cell cycle. At least you can force them to the first phase, like they go from G0 to G1 and then to S. But then they tend to get stuck, because if you remember when you study cell cycle, is those cyclin chaperone different phase of the cell cycle, and then after they are done chaperoning the phase, they actually have to be degraded and for another set of cyclin to come in. So to be able to make this work, you have to be able to, in a time-sensitive way, express a gene, shut it down, express another one, shut it down in a particular order without any mistake. So that's one of the challenges. The other approach is people have used stem cells, and there have been some clinical trial in the US and Germany, mostly in Germany, where they try to identify stem cell from different part of the body. So sometimes people have identified at first stem cell they believe are in the heart, and then they can cultivate them and reintroduce them into the heart. A lot of those have not had very clear results. What we know, let's say if you take stem cell from bone marrow, and there's been both animal studies and clinical trial where you can inject bone marrow cell into people that have had a heart attack, and then some of those bone marrow cells are going to home to the heart, and then they have noticed or they have documented that functionally the heart is doing better. But if you look at those cells, they actually don't become cardiac cells. So they can release factors in a paracrine manner that is going to make the heart function better, but um, without making new cardiac cell. There's also the caveat which happened in the German studies that sometimes when you inject stem cell, you can't really fully control their differentiation. So sometimes they actually differentiate into the kind of tissue that you don't want in the heart, maybe like bone, for example, right? So there's way to think about this. There are way to study and try to make this work, but we still have a lot of challenges that we have to overcome. Does that answer your question?
Thank you.
Sir, for a learning purpose, I have a question for Dr. Suresh, sir. Sir, what are the limitations of organoid models when compared to the in vivo models?
So, organoid models are in vitro models, in vivo models are animal models. So, obvious limitations are the physiology of the animal cannot be repeated in the organoid. Organoid is a static cell structure, that's a 3D structure. The best advantage is it's coming from the human cell. So it's a human organoid static comparing to an animal cell in an animal with an advantage of physiology or the flow. So the interface between these two is the microphysiological system or the organ on chip, where you can put multiple organoids on the plate and create a flow pattern between them just like the body and understand the physiology aspect. So there are multiple advantages for in vivo and disadvantages for organoid models. The best advantage is organoids are human relevant, animal models are animal relevant. Thank you.
I request you to make organize any workshops in this. If virtual also, it will be beneficial for.
Sure, yeah. You can come. We'll charge you some money for that.
Okay, these questions for our Sri Lankan experts. So in the lunch also we discussed about few. So I think this is good opportunity to talk about this. So could you please explain what are the current regulations and ethical guidelines for the use of animals in laboratory science?
Yes, actually, in 2008 to 9, we published a guideline, right? And there was no changes, and it was applicable for about 10 years. Now, very recently, SLALAS was invited by the NSF, that is the National Science Foundation, to develop a comprehensive guideline for the use of animals in research. So it was published and it's available. I think it is available in the website, our SLALAS website. So that is the guideline. At the moment, we don't have the welfare bill, right? So we are waiting for the enactment of the welfare bill.
Good afternoon to all. My question is, what are the best practices to manage the drop in and drop out during the studies? Because sometimes when we are doing the study, so our animals, rat or mice, get ill during the studies and we have to drop them out. So how can we manage this during our studies? Or because if we are including other animals into our studies during the meantime, so it will cause some biasness in the data, because half of the study is done and now the rat are getting ill or having any infectious disease. So we are including other animals, so just like drop in. So how can we manage this? There's a drop in and drop out. It's a general question. I'm not stick to any professor to do that in particular.
Actually, I mean, when there are droppings, actually that interpretation part, there will be problem. But at the initial level, you have to do the proper statistical analysis and you have to keep some rooms to, I mean, by unexpected variables. If you calculate that at the beginning, those problems can be solved. I don't know whether I have accurately answered to your question. That is the statistical part we do. I think some
sample size, right? So there can be instances where we have to go for 25% as well, depending on the study that we do.
And sometimes we are having that long study of 3 months and 6 months. In that type of study, we can do also like the 25% and.
Yes. Yeah.
And one more doubt that, like that, ma'am. Sometimes we write the application for getting 30 animals or 40 animals. During the study, especially related to neuro related study or cancer type, there are high morbidities in the during the study time period. So what problem we are facing when we are applying for the application again? It take more time. So is there any guidelines to get the animals to and complete the studies in that time? Because in between, if there is more death occurs, so we cannot continue.
So you have to apply that there is a 10% mortality, I think. Yeah.
Is your increase in the number. I think that way you can justify your request for an increased number of animals, and then you will not have a problem with the ethics.
Thank you so much, sir. Thank you.
Actually, this is an open question, and so when we evaluating an anti-fertility for a herbal plant extract on a zebrafish, so there is a many external factors that affecting that study. So the study is limited. So are there any alternative methods or any way to prevent that external effects on that fertility? But there is so many external factors that limit that study, right? Like sometimes the fish don't sperming. So how can we differentiate that this is the fertility effect or? Yeah.
human infertility drug? Oh, you. Yes, yes. Yes. So you want to develop a therapeutic to prevent like infertility, right? To prevent infertility. So your model is zebrafish. So the issue is that there are a lot of epigenetic and environmental factors. So as Madam said, one thing, of course, fish do sperm, right? But the issue is in zebrafish, it's external fertilization, right? So your question is to find alternative model because you don't want to use mice or rat models, right? So the issue is you don't have to stick to zebrafish all the time. So there are fish which has internal fertilization as well. Okay, so you can do some literature survey and find out which model is suitable for your study. Not just because zebrafish is available for the convenience. I think it's not the suitable model, but if you are not going to fertility per se, right, like whether it's going to fertile or not. Okay, good luck.
Very good afternoon, professors. Actually, nowadays we can see the numerous herbal compounds have been studied for various disorders. But I want to know what is the challenge in taking the preclinical studies into the clinical, and why I am asking this for the last 3 months I am searching for literatures regarding the Cosmium fenestratum which is available in the Sri Lanka. We want to work in that actually, but Cosmium fenestratum is they are using for long from the ancient times in the Sri Lanka, but I am checking for that no products available in Cosmium fenestratum. But we are getting the bark, the stem, but the products is not available. What is the challenge of taking the preclinical studies to clinical products?
For my clarification, your question is, though we do herbal experiment, there are no product development. Is that the question?
We are not applying, but just we are reporting. But what is the application of that?
Yeah, that is a question actually. That is a big question in Sri Lanka. Yes, there are a lot of animal experiments are going on, but mainly those are related to herbal compounds. Actually, most of the herbal compounds are time tested. More than 2,500 years they have been tested on human beings. There are controversies whether it is really needed to be tested on animals. That is in one hand. But in the allopathic system of medicine, if there are no evidence-based research, I mean, those things are hearsay. So because of that, there is a requirement to do animal experiment as well. And current scenario is, if you have published the animal data in a scientifically, recognized journal, you can go ahead with the work with the healthy human being, preclinical, that is, if you have published your data with the healthy volunteers, and then you can step into clinical trial. But it is very difficult to get the ethical clearance from the committees, because allopathic system and indigenous system, that not there are problems. So because of that, only very few animal experiments have been tested by using human beings, mean clinical trials, because it is difficult to get the ethical approval in this country. But there are some studies being done and published. Did I answer to your question?
Yes, ma'am, I got the answer. Now we are planning to do work with the use of the Coccineum fenestratum. This is a Sri Lankan plant, I think. Why I am telling you?
Coccineum fenestratum.
Yes,
okay, that is very well. Yeah.
Yes, many literatures reporting that Coccineum fenestratum having the more potential activities. Like after delivery, women are taking for increasing the blood count. Many things are there, productive effects are there. Now we are planning to work in Coccineum fenestratum. I hope next conference we will present that work here.
Congratulations and all the best. Yes, and doing clinical trials and stopping at that point is also worthless unless you can go into a product that will benefit the general public finally. So try as much as possible to go for some products, even though the trials are done, you will find that getting ethical clearance and doing the trial is much easier than finding a commercial partner to commercialize your product. So it's a long journey, but it's not impossible.
Thank you very much, everyone who participated in this engaging Q&A session. A special thank you goes to all the students and attendees who asked thoughtful and insightful questions. Your curiosity and enthusiasm have truly enriched this discussion. We also extend our heartfelt gratitude to our distinguished speakers and professors for taking time to provide such detailed and enlightening answers. Your expertise and willingness to share knowledge have made this session incredibly valuable for everyone present. Thank you very much, everyone, once again. With that, we now move on to the continuation of the scientific paper session, which will be chaired by Dr. Patrick Sharp, the Executive Director of Veterinary Services, University of California, USA. During this session, we will hear presentations of scientific papers advancing laboratory animal science. Now, I would like to invite Dr. Patrick Sharp, the Executive Director of Veterinary Services from University of California, to chair the session. Over to you, sir.
Okay. Our first talk will be assessing the toxicity of a novel dermatologic formulation reinforced with silver-decorated carbon nanostructures by YMPN Yapa. Hopefully I pronounced that correctly.
In the toxicity of a novel dermatological formulation reinforced with silver-decorated carbon nanostructures. So first of all, I would like to discuss with you about this fascinating technology, nanotechnology, especially in biomedical solutions. I think all of us know this nanotechnology offers promising solutions to various challenges in the biomedical field. And also when we talk about this nanotechnology, metal nanoparticles play a crucial role because they have unique physical and chemical properties. And among these unique physical and chemical properties, the generation of reactive oxygen species plays a crucial role here. And when we talk about the biomedical applications, the antimicrobial applications also play a significant role. And when we talk about the nanoparticles, due to their small size, actually they are very small, and due to the high surface area to volume ratio, it allows them to effectively interact with microbial cells and disrupt their function. And here you can see there are so many advantages of using metallic nanoparticles over the conventional treatment agents which are available in the commercial market. So they can reduce the antimicrobial resistance, they can have a synergistic activity when we use them in combinations. The efficiency is very high and the control release can be obtained, and also they are active against a broad spectrum of microbes. And when we talk about these metallic nanoparticles, silver nanoparticles are extremely important because so many researchers have proved they have a superior activity, antibacterial activity, antiviral activity, and antifungal activity from the silver nanoparticles. But do you know the superior antibacterial action can be hindered because of disadvantages. This disadvantage is the nanotoxicity of the silver nanoparticles. So actually I have aforementioned that silver nanoparticles is very tiny and they have a high surface to volume ratio. So this can generate reactive oxygen species which can be very toxic to the human cells and also the biological cells. But don't worry, we have so many strategies to reduce this toxicity. So one of those strategies is the doping of the silver nanoparticles into apparently non-toxic particles like carbon nanoparticles. So why you use carbon nanoparticles as the doping agent to dope the silver nanoparticles? Because it has a high surface area and also the pore diameter is homogeneous, the porosity is very high, and they are less toxicity and apparently biocompatible, and also it can act as carriers. And this pore network can be doped with various metals, and also the most important point thing here is they can lower the band gap energy. So what is this band gap energy? As you can see here in this diagram, the energy difference between the conductive band and the valence band called the band gap energy. And when we dope the silver nanoparticles into carbon nanoparticles, this band gap energy can be reduced, and also the wavelength which is active here is belong to the visible light region. And as you can see here in this diagram, there's a significant portion of visible light in the solar spectrum. So when we exposed this nanohybrid system to the visible light, it generated the reactive oxygen species under the normal conditions. So we don't have to provide any sophistical technologies here. So then it generated reactive oxygen species and then destroyed microbes by this generated reactive oxygen species. And this activity is also called as the self-sterilization ability of metallic nanoparticles. So these are our objectives of our study. First, to formulate the nanogel preparation by incorporated the silver doped carbon nanoparticles as an antimicrobial agent. Then to evaluate the antimicrobial potential of the nanogel preparations. And finally to assess the LD50 value of the nanoparticle systems and assess the skin irritation potential, skin diffusion potential, and finally the cytotoxicity. So here you can see our methodology. Actually, first we have synthesized silver nanoparticles and carbon nanoparticles. Carbon nanoparticles were synthesized by coconut coils, and we doped silver nanoparticles into these carbon nanoparticles by salt gel method. We characterized and finally we evaluated the toxicity potential of this nanogel preparation. Let's move on to the results and discussion session. Actually, I don't go to explain about the characterization of the nanoparticles. Here, I only focus to the antimicrobial effects and the toxicity effects of these nanogel preparations. So here you can see the scanning electron microscopic images and the transmission electron microscopic images. Here you can see the morphological characteristics, and also the despacing value is well matched with the XRD data. So this confirms successful synthesis of silver doped carbon nanoparticles and also successful doping of the silver nanoparticles into the carbon nanoparticles. So after this confirmation, we have evaluated the LC50 values for the synthesized nanohybrids. Here we have used zebrafish embryo test, and according to the OECD guidelines, and but we have used slight modifications. And here we have evaluated LC50 values for silver nanoparticles, carbon nanoparticles, and silver doped carbon nanoparticles. Actually, and we compared those values, and also additionally we have observed 4 apical points including coagulation, lack of somite formation, non-detachment of the tail, and lack of the heartbeat. So here you can see our evaluated LC50 values. So you can see silver dope carbon nanoparticles has the highest LD50 value, which means it has the lowest toxic effects when comparing with the silver nanoparticles alone. So this means a high dose of the silver dope carbon nanoparticles can be employed as an antimicrobial agent without lethal toxic effects when comparing with employed silver nanoparticles alone. And when we do the statistical data by the t-test, here also you can see that there is no significant difference between the LC50 values of carbon nanoparticles and silver dope carbon nanoparticles. This also confirms the doping of the silver nanoparticles into the carbon nanoparticle system significantly reduce the toxic effects of the silver nanoparticles. And here I have included some images of the zebrafish embryo toxicity assay. Here you can see there are so many abnormalities and coagulation of the eggs in silver nanoparticle sample. I have observed them up to 96 hours, and I have used 7 concentrations. And when it comes to the carbon nanoparticles and silver dope carbon nanoparticles, you can see there is a significantly reduction of these abnormalities when we dope the silver nanoparticles into the carbon nanoparticles. So then we assess in the apical points of the zebrafish embryo assay. Here also we have compared these 2 nanohybrids, and we have done the statistical analysis by Wilcoxon sign rank test. Here also, according to the statistical data, there is no significant difference between the negative control and the lower concentrations of the nanohybrids, but except silver nanoparticles. You can see there is significant difference between silver nanoparticles even in lower concentration. So this also confirms that using silver nanoparticles alone results a high toxicity when comparing with silver dope carbon nanoparticles. Then we formulated our nanogel preparation. Actually, you can see here we have prepared 3 type of nanogel preparations. They are silver nanoparticles incorporated one, carbon nanoparticles incorporated one, and silver nanoparticles incorporated carbon one. And I have included some images here also for your reference. And here we have evaluated the antimicrobial assays. In the number 4th row, you can see the silver dope carbon nanoparticles reduces the lowest IC50 value, which means it has the highest antimicrobial potential. And when we observe the MIC and MBC values also, silver dope carbon nanoparticles recorded the lowest MIC and MBC values here. So it also confirms the best formulation is silver dope carbon nanoparticles. And then we conducted the cytotoxicity assay of the nanogel by live and dead cell assay. Here we have used fluorescence microscoping imaging in day 1 and day 7. Here also, when we compare the results, the highest cell survival percentage was showed by silver dope carbon nanoparticles. And according to the statistical data, there are no significant difference between the negative control and the samples of silver nanoparticles and silver dope carbon nanoparticles. And then we evaluated the skin irritation potential, because I have aforementioned this is a dermatological application. So here we have used the headcam assay, and you can see our observations here. Actually, in silver dope carbon nanoparticles, there was no significant irritation observations like coagulation, lysis, or hemorrhages. And according to the total irritation score calculation, we have these irritation scores. And here also you can see the silver dope carbon nanoparticles has a very low irritation score, where the silver nanoparticles has a high irritation score. And the statistical data also shows that there are no significant difference between the negative control and the samples of silver dope carbon nanoparticles. And finally, we have conducted the skin diffusion assay by France diffusion cell, because we have to evaluate the skin permeation ability of this nanogel preparation. For this purpose, we have used the France diffusion cell assay. Here you can see the France diffusion cell which is used in our laboratory. And these are the results. You can see here silver dope carbon nanoparticles didn't show a significant penetration from the skin. And these are some of our recommendations. Considering the aforementioned results, it is recommended to conduct in vivo animal studies to further confirm this concept. And also the nanogel preparation which we developed here can be used as a topical application for various microbial infections, as a catheter gel, or as an oral application for a buccal cavity infections. So we can conclude at our research that it is confirmed that the silver dope carbon nanoparticles has the best antimicrobial activity, and doping the silver nanoparticles into carbon nanoparticles reduce the toxicity effects of silver nanoparticles while enhancing the antimicrobial properties. Thank you very much.
Thank you. Are there any questions?
There was no animal study done in this experiment. Your experiment is very good. Everything is in vitro, right?
Yes, actually, this is the initial state of our research. We have applied for the the ethical approval for the animal study. So that's why I put it as a recommendations. The nanogel is really working. Actually, I have a supporting slide to show the fascinating antimicrobial action of our nanogel preparation. So then the next step is the animal study. Thank you, sir.
I'm very convinced that it is working, but in vivo is the next
Yeah.
step, not part of this.
Yeah, thank you.
Thank you. Are there any other questions?
Molecule was used as a negative control.
Yes, sir.
And what was the diluent for the silver doped and carbon doped nanoparticles?
So actually we have used dispersions. The maximum concentration was 10 milligrams per liter.
No, I'm asking what was the diluent?
We didn't use a diluent, sir, because we can't use such chemicals because silver nanoparticles tend to be react with those. That's why we have used molecule water as the negative control also, because according to the guideline we have to use system water as the negative control. But in this case of nanoparticles, they have a tendency to react with those minerals in those water. So that's why we only used molecule water for prepare these dispersions.
Any other questions? Okay, thank you very much.
Thank you.
May I have your attention, please, before going into the next oral presenter? About the time allocation for your presentation, you are allocated with 10 minutes to present your presentation. And during this 10 minutes, one bell will be rang at the 8-minute mark. And there will be 2 bells once you are done, which means your 10 minutes is up. I hope that's clear to you all, and good luck.
Okay, the next presentation will be evaluation of cytotoxicity by allium-cepa cytotoxicity assay and identification of the active compounds in the leaves of Cyclea peltata. The presenter is APA Tashani. Again, apologies if that's incorrectly pronounced. So let's go to the next presentation, I've been advised. So the next is anti-obesity effects of Tamarindus indica, tamarind seed kernel, powder in Daniel feed with short-term high-fat diet. I'm sorry.
A healthy outside started from the inside. So my dear audience, let me introduce about my research. Anti-obesity effects of tamarind seed kernel powder in short-term high-fat diet-fed zebrafish. Here's the content of this presentation and let's move on to the introduction. Obesity, the real pandemic in 21st century, and defined as abnormal or excessive body fat accumulation in the adipose tissue, mainly due to imbalance in food intake. Are there chemical drugs to treat obesity? Yes. However, most of the approved drugs have been withdrawn from the market due to their adverse health effects and limited long-term success. So, possibility of utilizing natural alternative sources from each herbal plants considered as the potential therapeutic modality. So, here's the tamarind come to the stage. Tamarind is a multi-purpose tropical fruit species and popular for its medicinal properties at anti-inflammatory, anti-diabetic, and antimicrobial properties. And also, anti-obesogenic properties of tamarind fruit and seed corn are scientifically proven, but tamarind seed kernel is not yet tested for its anti-obesogenic properties. Tamarind seed kernel is the major part of the seed and composition is similar to the cereal seeds and make it suitable for human consumption and can be used in various food industries. But however, largely it is still considered as the waste or byproduct in the tamarind fruit industry. So as a value addition and to reduce the output as the waste and it interests to investigate anti-obesogenic potential of tamarind seed kernel powder and it did not yet scientifically proven. So the aim of the study was to investigate anti-obesogenic effects of tamarind seed kernel powder by using diet-induced obese zebrafish. Zebrafish is the promising and alternative model organism for the obesity research as diet-induced obese zebrafish and obese mammals share the common pathophysiological pathways and it can be fed with different high-fat diets for a standard period to establish diet-induced obese zebrafish model. So, the overall objective of the study was to investigate anti-obesogenic properties of tamarind seed kernel powder using the obese zebrafish model. Specific objectives were to study the effects of tamarind seed kernel powder incorporated diet on body weight, body mass index, specific growth rate, hepatosomatic index, and obesity-induced liver and intestine histopathology, and peroxisome peripherate activated receptor gamma, PIPA gamma gene expression in short-term high-fat diet fed induced obese zebrafish. Let me explain you the methodology. Under the experimental design, there are 5 treatments as the high-fat diet plus tamarind seed kernel powder 5%, high-fat diet plus 10% tamarind seed kernel powder, and high-fat diet plus 15% tamarind seed kernel powder, and a control diet. High-fat diet made with the recipe that locally available feed ingredients, and finally made the lipid content for the 30%. And while the control diet having the lipid content of the 9%. And standard procedure was followed on to make the tamarind seed kernel, to obtain the tamarind seed kernel powder to make the T5, T10 and T15 diets. Proximate analysis was done for all the 5 feeds and preference test was carried out to check the zebrafish acceptance of the feeds. Animal model used here was the zebrafish of 28 days post fertilization of age and number of fish per tank was 15 for duration of the experimental period was the 8 weeks and all the treatments are run in triplicates and the feeding was adjusted to do 5% of the body weight as the daily ration. Under the measurements, the growth parameters, total body weight, body length, body mass index, specific growth rate, and hepatosomatic index, the total body weight and body length measured weekly by using randomly selected 10 zebrafish from each tank and BMI calculated using this equation. Specific growth rate was calculated by using this equation, and hepatosomatic index was calculated at the end of the experimental period by using the 3 fish from each treatment as the randomly used. The histopathology of liver and intestinal tissue was done. Hematoxylin and e-zinc staining was used and observed variation in the tissue structure histology from the normal histology. Finally, the peroxisome peripherate activated receptor gamma, PIPA gamma gene expression was done. PIPA gamma is the adipocyte differentiation factor that can be analyzed using the obesity research. So QRT-PCR was done with specific primers of the PIPA gamma and the housekeeping gene ACTB. All the statistical analysis was done using Minitab and Microsoft Excel softwares. First, data was tested for normal distribution, and once ensuring the normal distribution, one way ANOVA with 2 key pairwise comparison was done. Paired t-tests also performed to ensure the significant difference of BMI over time in 5 treatments with the level of significance of 5%. Let me explain you the results and the discussion. Growth parameters are used as the most popular parameters in obesity research as body length, body weight, and BMI. So, for the body length, none of the feed types had a significant effects on body weight, and this result agreed with the previous diet-induced obese studies. And for the body weight, the highest body weight was obtained by high-fat diet fed zebrafish, followed by T5, T10, T15, and control diets. At the end of the experimental period, they were all significantly different from each other, and with the increase in tamarind kernel powder inclusions, significant difference in body weight can be seen. Then the body mass index, the highest body mass index was obtained in high-fat diet, followed by T5, T10, T15, and control diets. And at the end of the experimental period, they were significantly different from each other. And significantly high BMI under the HFD is indicated of successful establishment of obese zebrafish model. So with increase in tamarind kernel powder, there was a significant decrease in BMI. Here's the morphology of zebrafish at the end of the experimental period, and the results of weight and the BMI was ensured by the morphology. Liver is the primary organ that can get affected by the dietary lipids. So the hepatosomatic index is a good indicator to analyze the vital organs as the liver. So hepatosomatic index was highest in high-fat diet fed fish, followed by T5, T10, and they were significantly different from the T15 and control groups. So increase in tamarind kernel powder inclusions, there was a significant decrease in zebrafish hepatosomatic index. And furthermore, histological analysis of liver and intestinal tissue was done. Here, histological analysis of liver has been observed. Here, the high-fat diet fed fish liver, there are necrosis, vacuolization, and lipid accumulations. Those are the signs of obesity-induced liver damages. And here, the T15 diet fed fish liver, and here the normal control diet fed fish liver. So, as you can see here, the similar morphology in the T15 diet fed fish liver as the normal histology. I had done the intestinal histopathology, but with the time limitation, I had to omit it. And adipocyte differentiation lead to obesity. So, as the PIPA gamma is the adipocyte differentiation factor, it is a crucial factor and an important indicator to analyze the obesity in the molecular level. So, PIPA gamma gene expression was done by using QRT-PCR. Here's the high-fat diet obtained the highest PIPA gamma gene expression, followed by T5 and T10, and they were significantly different from the T15 and the control groups. So, T15 diet anti-adipogenic property was observed with the reduced adipocyte differentiation. Finally, those are the conclusion of the study. Fed in zebrafish with 30% of high-fat diet successfully induced diet-induced obesity in zebrafish. The inclusions of tamarind seed kernel powder with a high-fat diet lowered the total body weight, lowered the body mass index, lowered the hepatosomatic index, lowered the PIPA gamma gene expression, and finally reduced the lipid-induced histological changes in liver and intestine of zebrafish in dose-dependent manner. So T15, or the 15% of tamarind seed kernel powder inclusion, was shown to be most effective and can be proposed as anti-obesogenic agent. Those are the references I had followed, and thank you.
Thank you. Are there any questions?
Thank you. That's a nice study. I was wondering, there are different type of controls. Have you thought about doing groups where you have the control feeding plus tamarind extract, plus tamarind powder?
Sir, here I want to find out the anti-adipogenic effect of the tamarind seed kernel powder. So I used the inclusions with high fat diet. First, I give high fat diet to one group, and with the high fat diet, I use T5, T10, and T15, and finally a control diet to see whether is there any the environmental conditions or what happened, or any unexpected happen, or other things happen to these diets, or any other external factor affect to this zebrafish.
So I guess I was wondering, if you were to use normal diet plus tamarind powder, would you expect a decrease in weight? Might you be able to figure out like the mechanism? Why is it anti-obesity? Is it because it interferes with absorption in the guts, or is it something else?
With my findings, I guess that tamarind seed kernel powder having more bioactive compounds. They having many phytochemicals and other trypsin inhibitors and tannins and other bioactive compounds. So with the compounds, there is a inhibition of the obesity or the adipocyte differentiation, and the cell signaling pathways like insulin signaling pathways and inflammatory pathways, they also can be affected with the reducing weight and the obesity.
Thank you. I would continue that very good question from Dr. Pascal. I think in your experiment, you are looking at the aquatic animal and adipose and extrapolating the result to a terrestrial usage. I don't know I think you have to definitely do the in vivo experiments. But more importantly, from Dr. Pascal's question, the mechanism of action is very critical. And going back to, I think, Srinath's question on why these herbal compounds are not getting treated like allopathic compounds, the main reason for that is the active ingredient and the mechanism of action of herbal compounds are not clearly shown, and that's why they go into a different track. So if you want to pursue this, because there's you're missing a picture of the tamarind kernel for general audience. I have seen it. So if you are pursuing that one, I would like you to show the anti-insulin dependent obesity reduction, because insulin, diabetes, obesity are closely linked, and liver changes in a terrestrial mammal like rat or mouse or rabbit or dog or monkey, and then take it forward for identifying the active ingredient rather than a whole kernel powder. That will not get approved as a allopathic compound or an active ingredient. So it's important for you to show the mechanism of action. Good work so far, and a nice model selection.
Thank you, sir. Thank you. Yes.
Any other questions?
So it's very nice and interesting presentation. Thank you very
madam.
much. I would like to know the zebrafish that you have used in this study. Are they males, females, or both?
Madam, I selected the juvenile zebrafish at 8 days post-fertilization. So at that age, we can't identify as males or females. And I ended my experiment like in their juvenile stage 2. So I can't differentiate they are males or females, but I used the juvenile stage just because the metabolic rate is very high than the adults, and their metabolic pathways, they mimicry the human obesity as well. That's why using juvenile zebrafish. Yes, so I can't differentiate they are males or females.
So 28 days is just 1 month old zebrafish, but your pictures looks a little bit older than that.
It was taken like end of the experimental period, like in 80 days, so 85 days.
85 days.
Yeah, 80.
And the other one, yes, there's a. I'll give it to you. Wait a minute. So another question is that when you do this study, when it comes to the end, you said they are a little bit mature, right? Up to maturity. But it's better to take one sex. Like I'm becoming sex biased, but avoid females because in your picture, I saw female fish, most of them. So female fish, maturity, their belly usually. But I'm really happy that in your study you have done the gene analysis and histopathology and proved there's an effect. But please consider that factor as well.
Sure, madam.
I have one more question. Nice presentation and
Thank you, madam.
For my information, what is the source of this zebrafish? From where did you get this zebrafish juveniles?
We bought this fish from the commercial breeder, like in Nitabur or some area, and took to our laboratories and done the experiment.
Right. Thank you.
Thank you, madam.
Any other questions? All right, thank you very much.
Thank you.
The next presentation will be enhancing preclinical drug screening with tumoroid models, the OncoSeq bio approach. I think, yeah.
Yes. Good morning, everybody. My name is Reshma, and I'm from OncoSeq Bio. Here today, I'm going to present on the topic enhancing preclinical drug screening with tumoroid models, the OncoSeq Bio approach. So, people here were talking about so much on the drug screening process in the clinical studies. Here, we are working morely on the preclinical studies. I'll give a brief explanation. Like there is a for the preclinical drug screening methodology. There we will go for the in vitro station. In vitro drug screening, we will usually go for the 2D. That's a monolayer. In our expertise, we used to go for the 3D cell cultures, which we will be deriving the cells from the stem cells, cell lines, and the patient-derived. So the patient-derived cells are called tumoroid models. Why we are actually going for the 3D cell culture models? Because it has the 3D architecture and the cellular heterogeneity, which will be replicating our human physiology, which is in our body. It is close to human, and the microenvironment which we will be providing while we are culturing the cells. Here we are following the 3R principles by reduce, refine, and replace. By reducing the number of animals in the preclinical drug screening methodology and refine it with the minimal stress by enhancing in the tumoroid models. And we are replacing with the alternative methodologies like MPS, which we'll be seeing further. So we know the traditional methodologies which we are using for the like centuries right now, and the next the modern day models are the spheroids, organoids, and the transfer methods. Now the advanced technology which we are will be using is a next generation models, which is body on chip, like 3D bioprinting and the modeling immunotherapy, the iPSC, like stem cells derived for the organoid development. So these models are called as the microphysiological system, which was asked previously in the questions actually. So, and why we are actually doing it, because it's we are creating a disease model and which replicates how which is actually inside our body. So we can replicate what is the disease which is actually happening and the function it is. And we can, so that we can give the precision medicine and the clinical transplant. So, and we have the wide range of the cancer cell lines, which is prostate, colon, liver, glioblastoma, and lung cancer, breast cancer we have. And we have standardized the spheroid culture in our lab so that we can, in different types. So we can, this forms the disease models or the cancer types, tumoroid models, and we can use it for the further drug screening methodologies. Here is a simple example I'm giving. This is a confocal microscopy of the 3D spheroids of the DU145 of the spheroid before the treatment and after the treatment. Here you can see the spheroid which is being formed in the integrated structure, and here it is a cell bulbing has been happening after the doxorubicin. Doxorubicin is an anti-cancer drug which is widely used. So on the IC50 value which is being treated on the DU145, you can see the cell bulbing has been happening over here. I can show you. Yes. This is a Z stacking. Z stacking is something like in the 3D models, you can't see how it is actually happening. So for analysis, how the cellular structures have been happening, we go for the Z stacking in the confocal imaging systems. So on this system, like you can see on the layers, how this is a control. It is in the integrated form, and you can see how it is disintegrating. The rhodamin actually sticks to the cytoskeleton, and you can see the blue color is a staining of the nucleus. The nucleus stain has been decreased over here because the cells are going under the cytoskeleton. I mean, well-being has been happening over here. The next, we have also developed a disease model which is called NASH model. So NASH is non-fatty acid. Non-fatty, non-alcoholic fatty liver acid. So it's fatty acid induced spheroid model have been successfully shows the lipid accumulation in the series of days like day 2, day 4, and day 6 by staining the Nile red staining. It's a lipophilic staining which stains in the fatty acids, and you can see we have developed the fatty livers with the co-cultures of the 3 different cell lines. Over here you can see, and this is a clear explanation of the how the we want to study how the cellular levels will be there in the spheroid models. So here we have given the rhodamine over here for the cytoskeleton staining and the DAPI. This is a Z stacking. So at each level, how it will be, we can study in the levels. It is different on different channels we are capturing, and we are composite over here. You can see it's a red channel and the bright field channel and the DAPI channel. It's a composite over here. So this is we also do the imaging analysis with the IXMCs, which is a high-end imaging system. With that, you can see how we are creating the spheroid models. It's a day at the 0th hour, and we are creating a model. You can see we have actually tracked down, like we have stained the cells with a green cell tracker so that we can able to observe how it is going on. So this is the cells at the 0th hour, and here we used the cell as a ovarian cancer cell line, which is ES2. And over the 48 hours, it has been formed successfully as a spheroid, and in the we are capturing under the bright field and the DAPI channels to observe how is it. We also having we are also doing the co-culture models with the NK cells, which you can actually see in Ramya's poster over there. It's on the 48 hours, the cells is there, and we are actually co-culturing with the NK cells over here. You can see the surrounding cells, which is like a dot, which are NK cells. And how it is, we want to see how it is actually reacting also, because it is a 3D cell culture model, and how it is going, affecting its immune cells. So we actually tagged the NK cells with a green cell tracker, and we were analyzing how it is going on. It's a pH staining, and over here, like, cells are actually dying. It's a composite cell line. You can see how it is entering and killing all the cells around, and how it is going on. This is the cell level of studies which we are doing right now. So here the oncosic bio approach is like, we are doing the comprehensive tumoroid and organoids platform for the precision medicine, actually, to accelerate the efficient preclinical testing. We are doing the ethical approach, reducing the animal usages, expertise in the integrating advancing analyzing tools. We also support the collaborative research in the studies of the preclinical cancer research. Over here, why from the preclinicals to the clinical success, how it actually goes. Here it's a tumoroid bridges the gap between the in vitro and the clinical outcomes, because we will be getting the cells directly from the patient, and we will be forming the tumoroid, which is a 3D cell culture. We will be testing our drugs on that one. So we can see how it is reacting, and we can analyze what will be the precision medicine for the particular patients. So here provides the accurate platform for identifying effective cancer therapies over here. And it's like a seamless transition from the lab research to the clinical applications we can go, and which empowers the researchers and the clinicians with the tools to enhance therapeutics outcomes to accelerate the journey towards the precision medicine. And this is a step towards the personalized cancer care by deriving the patient-derived tumor cells, and predicting the specific patient response, and facilitating the tailored therapeutic strategies. People can scan the QR code, and we can visit the website and have the more information about it.
Are there any questions?
I have a couple. The first one, I'm curious, in the fatty liver spheroid, what 3 cell type that you have. The second one is, are there specific particular time frame when you have to use your spheroid so they are kind of synchronized? And then the third one has to do with doxorubicin, which is known to actually cause cardiac myopathy. So
Yes.
have you done any study with, I guess,
No,
cardiac
sir.
muscle
We
steroid?
have, first I'll answer from the questions. We have multiple cell lines, which we will be using according to the studies which we have done so far. And the second question, what was the second question, sir?
The first one was type of cell you use for the fatty liver.
Fi-
The fatty liver.
Fatty liver,
model.
which is liver HepG2 cells and stellate cells, and kidney endothelial cells. It's a co-culture of 3 cells together. Has a co-culture model. As I showed you, immuno co-culture model. That was a previously disease model which we produced. So the next question
What's the time window? So let's say you create those organoids, and they're gonna grow, right? And
Yes.
they're gonna reorganize.
Yes.
Is there a point at which that's optimal for use?
Yes.
And a point beyond which that shouldn't be used?
Yes, sir. So each cells behave differently, actually. So one cell will be obtaining. Each cells have the half-life time in the different. We know it. So in the same ways, each cell, like, from, you can observe after the 48 hours up to the 5 days of time period, and we can optimize and standardize when it is optimum, when there is a necrotic zone is 0. The necrotic zone is like when you can see it over here. Can I go? So you can see over here the central zone is called necrotic zone. The center, like it is 0 necrotic zone. We optimized it where the necrotic zone is like 0 or a very minimal. We can't see the cells where there is 0 necrotic zone. So when there is a minimal of necrotic zone, that is when the optimized condition it has been. So that is the time we can analyze our drugs on that.
Any other questions? I have a question. So when you're doing your work with the cell lines, how do you accommodate for authenticity of the cell lines? How do you test for authenticity? And then like for contamination? How do you assess?
I think we actually check the cells for genotyping just before.
Okay.
And also for contamination, we do the pathogens.
So you're looking for principally mycoplasma or?
Yes.
Gotcha. I figured you would, but just thought I'd ask. Okay, thank you. Any other questions? Okay, thank you very much.
Thank you.
Sorry, so we're going to go again with the evaluation of cytotoxicity by allium SEPA assay and identification of the active compounds in the leaves of Celia pautata.
Good afternoon, respected guests, researchers, and all the participants of the 12th Annual Scientific Session and International Conference. I'm Ayodhya Teshani, and I'm excited to present our research study, Evaluation of Cytotoxicity by Allium SEPA Cytotoxicity Test and Phytochemicals of Aqueous Extract of Cyclia peltata leaves. This work was collaborated with my colleagues NMCD Navaratna, GP Dini Kavindya, and the guidance and the support by Mrs. Nirmani Samrakon and Mr. Aravinda Sirivardhana. And let me take you through our study to explore the cytotoxic effect of the Cyclia peltata aqueous extract and the identification of the active compounds which is responsible for its medicinal properties. Cyclia peltata. In Sinhala we call kahipittam. It is a climbing shrub which is mostly spread in tropical climate. So we can find it in Sri Lanka, India, Malaysia like countries. And primarily this is used in traditional medicine for its anti-inflammatory, analgesic and antipyretic properties. So when we got to know about this plant, we had some questions like, does Cyclia peltata harm cells in different dosages? What active compound does Cyclia peltata have? So to find the solution for that, we designed laboratory experiment to the aqueous extract of the fresh Cyclia peltata leaves. In here we use for the cytotoxicity test, allium SEPA root growth inhibition test
And for the identify the active compounds, we did phytochemical analysis. In phytochemical analysis, first we selected some common active compounds in medicinal plants, and then we did the test to identify whether they present or absent. In this table, I had mentioned the active compounds we used and the test method we followed. And for the cytotoxicity, we did allium cepa root growth inhibition assay. In this flow chart, I had mentioned the steps we followed. In briefly, we exposed the onion bulbs into the aqueous extract of the Cyclia peltata leaves for 96 hours. Within these 96 hours, key metrics like morphological changes and the root growth were recorded. And as the result of the phytochemical analysis, we got positive result for phenol, saponins, tannins, and reducing sugars, which is responsible for its traditional medicinal applications. And in cytotoxicity assay, we got concentration dependent toxicity. That means in lowest concentration, the root growth is higher, whereas in higher concentration, root growth is low. And we saw some necrosis, gel-like structure, and browning of roots as the morphological changes. And after that, those results were plotted in a graph. According to the graph, the root growth percentage is higher in lowest concentration, and in higher concentration, the root growth percentage is low. And then after that, we calculate the inhibition concentration. That means the concentration which inhibited the root growth by 50%. According to all the result of the experiment, this in cytotoxicity assay, it shows the highest concentration have toxic effect of the effect on the allium cepa roots. That is prevent the root cells division and the metabolic activities. So when using the aqueous extract of Cyclia peltata leaves, we have to consider about the dosage ranges, and in phytochemical analysis that shows the presence of phenols, saponins, tannins, and reducing sugars, which is responsible for its anti-inflammatory, antioxidant, antimicrobial activities. And our study lays the groundwork for further studies like quantitative analysis of active compounds and the comparative studies and the identification of anti-inflammatory properties like studies. As the conclusion, our study gives the insight to the safe dosage and the potential therapeutic effect in both modern and traditional medicine application of the aqueous extract of the Cyclia peltata leaves. And these are the references we follow. Before the end, I would like to say medicinal plants are nature's gift for the humanity, and they help in many health challenges. By protecting and preserving, we not only honor in the nature but also have possibility for future medical discoveries. Every plant we save today could lead life-saving treatment tomorrow. So let's committed to safeguarding these treasures for benefits of all. And thank you. And I would like to thank my colleagues, my supervisors, and for the KI University and for the SLALAS for giving this valuable opportunity for me and the support of them. And thank you for your attention, and I welcome any question or feedback. Thank you.
Thank you. Are there any questions? Okay, no questions. Okay, well, thank you very much. Sorry, we have a question. I don't have eyes behind my head.
Yes, thank you for the presentation. I just want to know, you have applied for the ERC approval for this study?
Yes, madam.
So, actually, why was that needed?
because when doing the research we have to obey their rules because overuse of these plants and conservation.
That is, from where did you get the ethical approval for the study?
This is our undergraduate project, research. So we have to take the ERC approval from the campus.
Okay.
Yes, madam.
Okay. So this is only a part of your
Yes, ma'am.
study.
Undergraduate.
All right. So I just want to suggest now you have done this morphometric analysis of the growth of the roots, right?
Yes,
so you
ma'am.
can expand it further with the genotoxicity assays
Yes,
by
ma'am.
calculating mitotics index and everything. So you can expand and you can get a good collection of data if you just little expand it to genetic analysis as well. Okay, all the best. Thank you.
With that, we mark the end of the 12th annual. With that, we mark the end of the scientific paper session for the day 2 of 12th annual session and international conference of Sri Lanka Association for Laboratory Animal Science. And I would like to thank all the participants and the presenters as well as our panelists, including Dr. Patrick Sharp. Thank you everyone once again. Ladies and gentlemen, with that, we now take a moment to recognize the outstanding achievements of our participants with the awarding of the certificates for their contributions to the conference. We will begin by presenting the following awards. To start up, the Professor Vera Baumans Award. Emeritus Professor Vera Baumans, a specialist in laboratory animal science at Utrecht University, played a pivotal role in organizing the first international certificate course on laboratory animal science in Asia in 2011 at the University of Colombo. She was also the chief guest at SLALAS inaugural scientific session in 2014, where she was honored with the honorary membership for her global contributions. Professor Baumans nominated Professor Mangala Gunathilaka for the Charles River Prize in 2022, which led her becoming the first South Asian recipient of the prestigious award. In recognition of her support for the development of laboratory animal science in Sri Lanka, the Emeritus Professor Vera Baumans Award for the best publication in laboratory animal science was established. This award, funded by the interest on the portion of the Charles River Prize money, was first presented at SLALAS Annual Scientific Conference in January 2024 and is open to life members of SLALAS who have been members for at least 3 years. However, this year, none of the applications met the required criteria to qualify for the Emeritus Professor Vera Baumans Award for the best publication in laboratory animal science. And we do have 4 other awards in line. So these awards include Oncoseed Bio Award for the best poster presentation, Oncoseed Bio Award for the best oral presentation, and we have Dr. Shamini Jayasekara Award for the best oral presentation. I think we need a little bit more time in finalizing the final marks of these awards. At the meantime, we are going to present the certificate of appreciation for all the presenters in the annual conference of Sri Lanka Association for Laboratory Animal Science. For this, I would like to invite the chair of our panel, Dr. Patrick Sharp, onto stage. I'll be reading the names of the presenters of abstracts in the SLALAS session of SLALAS 12th annual session. Mukul Sham, Miss Piyumika Yapa, Mr. Pratap Srirangan, Miss S. Reshma, Miss VVS Ramya, Miss Shamina Laidari da Silva, B.M. Basnayaka, Miss Chamishka Uluwaduge, Miss W.G. Dinushi, Dr. Hasanti Ratnadivakara, NMCD Navaratna, LRIPK Vijay Ratna, Mr. Keisuke Masuda, Miss APA Teshani, Congratulations to all the presenters and thank you very much, Dr. Patrick Sharp. Thank you all for your patience as we approach the conclusion of today's program. At the moment, our esteemed judges are finalizing the marks and preparing the certificates for the award. And now we are going for the OncoSeek Bio Medal for the poster presentations, and we are going to start with the third place. Before that, let me give a moment to give the citation for the OncoSeek Bio Award. The OncoSeek Bio Awards, established in 2024, are generously sponsored and presented by Dr. Suresh Poosala, the founder and the president of OncoSeek Bio. Dr. Poosala, a long-time friend and a supporter of the Sri Lanka Association for Laboratory Animal Science, has been a dedicated scientist for advancement in the field of biomedical research. Through this initiative, Dr. Poosala aims to inspire and support the next generation of researchers while also strengthening the collaborative relationship between SLALAS and the broader scientific community. Now, I would like to invite Dr. Suresh Poosala to present the award for the OncoSeek Bio Award for the best poster presentations.
So thank you so much for entertaining my idea of supporting research in Sri Lanka. Judging today was not easy. Me, Patrick, and Shalahat were actually caught in two minds on deciding the first place, which was a very close call. The only reason why the first place got today's first prize was because he was an individual contributor to the entire research that we saw on the poster. It was not a collaborative effort. And for the second and third places, I think Patrick and we discussed all these awards and it was a close call. Chalawat, you are missing in that discussion. So it was a hard-fought battle for the best poster award, and we decided to give 3 awards instead of 2. So thanks to SLALAS for entertaining that idea as well.
Thank you very much, sir. With that, the most anticipated moment, the OncoSeek Bio sponsored award for the best poster presentation. The third presentation goes to BMPI Basnayaka for her abstract in a study on evaluating the impact of RIDE guidelines on the quality of zebra fish research. So the OncoSeek Bio sponsored award for the best poster presentation, the second place goes to Mr. Keisuke Masuda. And finally, the OncoSeek Bio sponsored award for the best poster presentation, the winner is Mr. Pratap Srirangan. We invite all 3 winners on stage to a group photo with Dr. Poosala. Could you please all the 3 winners come on stage? Yes. We would like to invite our judge board as well for this group photo. Dr. Lafontaine? Yeah. All right. Once again, we would like to invite Dr. Suresh Pusala. Now this is the time for the OncoSeek Bio Award for the Best Oral Presentation. We are delighted to inform you that there are 2 second place winners. OncoSeek Bio sponsored award for the best oral presentation. The 3rd place goes to Mr. Mukul Sham. OncoSeek Bio sponsored award for the best oral presentation. The 2nd place goes to Ms. Chamiska Uluwaduge. And finally, OncoSeek Bio sponsored award for the best oral presentation. 1st place goes to IPIPK Vijayaratna. And I would like to inform Mr. Mukul, you share the 2nd place with the Ms. Chamiska Udualuge. And once again, I would like to invite all the winners as well as Dr. Fusala and our judge board to take a group photo. Yes. Dr. Patrick Sharp, Dr. Arvind, and Dr. Cholawat. And finally, we have Shamini Jayasekara Award for the Best Oral Presentation. Let me read the citation for the Dr. Shamini Jayasekara Award. As the first laboratory animal veterinarian in Sri Lanka and the founding veterinarian at the Animal Center of the Medical Research Institute, from 1989 to 2010, she played a pivotal role in establishing animal colonies, producing feed, and making significant contributions to the field. In recognition of her invaluable work in laboratory animal science, this award has been named in her honor. She also generously sponsored this award in its early years, further supporting the advance of this discipline. With that, I would like to invite our chief guest, Professor Toru Takio, accompanied by the President of Sri Lanka Association for Laboratory Animal Science, Dr. Sachin Yamarasekara, to present the Shamini Jayasekara Award for the Best Oral Presentation and the cash prize. Ladies and gentlemen, Dr. Shamini Jayasekara Award for the Best Oral Presentation goes to LRIPK Vijayaratna. Thank you very much, Professor Toru and Dr. Sachini Amarasekara. And with that, I would like to invite the Secretary of SLALAS to deliver the vote of thanks. This is over to Ms. Piumi Kayapa.
Okay, good evening, all of you. We have finally come to the end of the 2 remarkable days in Sri Lanka's history. So, on behalf of the Sri Lanka Association for Laboratory Animal Science, I would like to express our sincere thanks to our chief guest, Professor Toru Tagio, for gracing us with his presence and valuable insights. It is truly an honor to have you here, sir. And we are deeply grateful to all our distinguished international speakers who have generously joined us from across the globe, Professor Cholavat Pacharinsak, Professor Suresh Pusala, Professor Marshall Frajbulat, Dr. Patrick Sharp, Dr. Satohiro Nakio, Dr. Aravind Ingle, and Professor Pascal J. Lafontaine. We truly appreciate your willingness to share your expertise and your time with us, enriching the discussions and offering invaluable perspectives. And also, I would like to extend my sincere gratitude to our local speakers as well, Professor Mangala Gunathilaka, Professor Sugandhika Suresh, and Dr. Mayuri Tammittiya Godege. And our heartfelt thanks to our sponsors whose support has been crucial in making this conference possible. A special thank you to ICLAS, our main sponsor, as well as our bronze sponsor, People's Bank Town Hall branch. And also, we would grateful to Helinta, RMC Ceylon, and Atlas Company for their generous contributions. I would also like to take this opportunity to thank all our special invitees, SLALAS members, and also the award winners of today. Your participation is vital to succeed this event, and also I would like to express my sincere gratitude to all the presenters of abstract presentations. Thank you very much for your presence today. And also our appreciation also extended to the staff of the Renuka City Hotel for their hospitality and assistance in ensuring that everything runs smoothly. We would also like to thank First Media for their excellent media coverage and photography, which will help us document and share the highlights of this event with a wider audience. And additionally, our heartfelt thanks go to the students from the University of Colombo for their invaluable support during the conference. Your dedication and assistance have been crucial to the success of this event, especially Ms. Sajini Dias, who compare whole event, and the other students as well, Ms. Nipuni Vanyarachi, Ms. Yashodhara, and Ms. Imasha. And also I would like to express my sincere gratitude to EOC Global for transport arrangements, and also to Helinta Products for the gift products, and RMC Ceylon for also for the gift products, and Ms. Maheshi Hennayaka on behalf of the Atlas Company, and for the printer products, University of Colombo Press, Pinterex, Printict, Flexport, Mime Sri Lanka, and Eco Product Sri Lanka. And also I would like to extend my sincere gratitude to the collaborators for this conference and for the pre-conference workshops. Special thanks goes to Medical Research Institute, Colombo, and Sri Lanka Veterinary Association, and CIMB, Department of Zoology, Environmental Science, Department of Zoology and Environmental Science, University of Colombo, and Professor Toru Takio from Kumamoto University, Japan. And also, I would like to extend my sincere gratitude to the 12th Executive Committee for all your efforts behind the scenes, and especially I would like to thank you, Dr. Sachini Amarasekara, the President of Sri Lanka Association for Laboratory Animal Science, your guidance and leadership for succeed this event. And also I would like to extend my sincere gratitude to Oncosic Bio Company India, Dr. Suresh Pusala. Thank you very much for all your sponsorships and for your support. So lastly, I would like to thank each and every one of you who has worked behind the scenes to ensure the success of this event. Your hard work and commitment have not unnoticed. So thank you all of you for your presence, support and contributions for this event, and let us continue to collaborate, share knowledge and work towards the advancement of laboratory animal science in Sri Lanka and beyond. Thank you very much.
Thank you very much, Ms. Piyumika Ayappa, for that heartfelt vote of thanks. As we come to the close of the 12th annual session and international conference of Sri Lanka Association for Laboratory Animal Science, we would like to extend our heartfelt gratitude to each one of you for your active participation, valuable contributions, and for making this conference a tremendous success. Over the past 2 days, we have shared knowledge, fostered collaborations, and built connections that will undoubtedly advance the field of laboratory animal science. With sincere thanks, all our esteemed speakers, panelists, participants, and volunteers for their dedication and efforts. Your presence and engagement have truly made this conference memorable. As we wrap up today's proceedings, we invite you to enjoy the tea and refreshments, which will be served outside the hall. We hope you will have a fulfilling experience, and we look forward to seeing you again in our future sessions. Today, we continue to shape the future of scientific research. Thank you once again. We wish you safe travels and see you at the next year's conference. And for the SLALAS members, we invite you to stay for the annual general meeting following the conclusion of the conference session. Once again, thank you all of you for your valuable participation and hope you enjoy the event. Thank you.